it's the last day of the Genome Design Analytics short course run by the UCSD team of the Center for Biosustainability. We're focusing on disseminating the computational tools we've been developing for strain design. The topic for today, the last day of it, is kinetic and thermodynamic modeling. It's just an overview of what we'll be going through today. Into presentation mode. So the first half of the class will be focused on what is in a kinetic model and how do you actually construct them. Also covering thermodynamics as another fundamental aspect of these models. And then we'll be covering these basic concepts in kinetic modeling. What kind of properties do kinetic models have that other models don't? What do we typically look for when we're analyzing a kinetic model? Covering reaction thermodynamics, the basics there, understanding where these constants come from, and then the ins and outs of dynamic simulation. So how do you set up a model? How do you graph a model in various ways to learn different things about the dynamics of your system? As an exercise, we'll be going through the MassPy Python package that was developed by Zach Heyman, who is in attendance today and will be leading the exercises. So that will be really nice. And then the second part will be covering specifically beyond reaction kinetics, how do we model enzyme kinetics, which have their own rules, and how we represent different enzyme mechanisms. How do we obtain the kinetic parameters for them? And then I'll be going through one case study, at least, in applying kinetic models to data analysis, which was this really nice paper that came out in Science about 5 years ago. Just really quickly, in terms of the materials we'll be going over today, they are in the GDGA short course public GitHub repository, which is located at GitHub, SPIG, which is our research group, and then / GDGA, Genome Design Genome Analytics _ short course. So it's a public repository. Everyone should be able to clone this, and you'll see the slides. You'll see, so the slides, you'll see the exercises, and you should be good to go. In terms of the package we'll be using today, that's the MassPy kinetic modeling package, located at this repository, GitHub /SPIG/MassPy. You can install it with pip. So if you haven't done that, I encourage you to try it. There's a Docker as well if you want to make sure that your environment is set up properly to use all the scripts. MassPy has really excellent documentation. And so if you miss anything, it's all here, but we'll be stepping through a lot of this material in the first exercise. There's also another repository that we've been developing in for strain design applications of kinetic models that is private currently, which is the Strainware K, but we'll be publishing examples from this shortly, and some of the the second exercise workflow, some of that workflow was inspired by the material here, so we're covering it in some form. Okay, so that's the repositories for today, and we'll just be stepping through these 3 notebooks: MassPy tutorial, model construction, then practical application in the exercises. All right, so let's get going then. So the first part of this class is going to be on building kinetic and thermodynamic models, what goes into these, and how they differ from the steady state models that we talked about in the first session. Just to go over a couple resources, I really like this Contemporary Enzyme Kinetics and Mechanisms book. This is based on, to what I understand, a series of papers published in Methods in Enzymology. They were collected and kind of concatenated into this textbook, but it covers all aspects of kinetic enzyme experimentation, so practical enzymology, as well as a lot of these trickier issues, like how do you model certain inhibition types? How do you model how kinetics change with pH and temperature? There's a lot of special topics in this book, so I always recommend that, which is available through a UCSD subscription. If you're here, you might be able to find it at your own institute. The other is iLab's own textbook. So Dr. Paulson has written 2 textbooks on systems biology. The first being on constraint-based modeling, which is one of the first textbooks on that topic. The second was this kinetic modeling follow-up, Systems Biology 2, Simulation of Dynamic Network States. That has been the source of a lot of the concepts in this lecture on how do you actually set up and analyze dynamic models. Okay, so let's get right into it then. The first concept here is to define our dynamic mass balances, because this is the the basis for steady state models that we talked about in the first session. It's also the basis for dynamic models. So what we're dealing with here with the metabolic model is a reaction network, right? So we have a network of reactions. What we're looking at in panel A here is what we call a node map, meaning that we're focusing on a particular metabolite, and we're showing the reactions that either produce the metabolite, and so add to the pool, or consume the metabolite, and so remove the metabolite from the pool, reduce the pool. And this is just an alternative way of viewing a reaction network, is to view the metabolite pools, view it from a metabolite centric perspective. And we talked about already how you can formulate a mass balance describing the consumption and production of this metabolite. So we have here our differential equation, the change in this metabolite over time, and it's just the production fluxes that add to it minus the consumption fluxes, so minus each of the fluxes that consume it. And we already talked about how we can set up a stoichiometry for this reaction. So the stoichiometry is positive if they produce it, negative if they consume it, which gets multiplied by the flux values themselves to tell us whether, for a given set of fluxes, our metabolite pool is increasing or decreasing. Now, in the steady state models, we also said how we make the assumption that our metabolite pools are not changing. And so our dx dt is equal to 0. And instead of handling this like a differential equation, we can make a stoichiometric matrix and handle our system with constraint-based modeling instead, which is an entirely different mathematical way to model it. With kinetic models, though, we just don't make that additional assumption. We allow our metabolite pools to vary dynamically, and instead we model this with a differential equation system and try to simulate its properties over time or understand them at steady state. So just going into the content here of our dynamic mass balance now. We once again have our stoichiometric matrix multiplied by our fluxes. So what has changed? Well, one, instead of being S times V equals 0, which is what we had in our steady state modeling or constraint-based modeling formalism, we now have a dynamic system. So now it's a mass change or a molar change per volume per time. We still have our dimensionless stoichiometric matrix, and we still have our V here, but instead of V being a single value or a vector, it's still a vector, but it's a vector of functions. So now we have our V is a what we call a rate law. So it's a function here of our concentrations and our kinetic parameters, the concentrations being in mass or moles per volume, and then our rate constants being in per time units. So what this ends up looking like is something like this, where we have to define our system, we have to define our conditions, and then our data, so any kind of metabolite or enzyme concentration data, and we define our dynamic mass balances, and we end up with this kind of differential equation system. And now here we have two options. The simplest way to dynamically model our system is to actually solve this over time for some initial conditions. So we specify some kind of, so we've already specified our rate constants here just so that this system is solvable. And we define some initial conditions. So ideally conditions that are not steady state, because in steady state, this will be 0 and nothing will move. So if you define some non-steady conditions, let's say some kind of perturbation to your system away from steady state, you numerically solve your concentration profiles over time. You can graph the results. This is what we just call simulation. You can also just analyze the properties of your model at steady state. So for example, you can calculate derivatives, the changes in your rate laws with respect to some parameters, dV dx, for example, and identify which fluxes are most sensitive to which metabolites. You can calculate things like the eigenvalues of your system to figure out if it's stable. You can do different analyses to figure out things like time constants, pools. And all of these are valuable to do in and of themselves, but today we're mainly going to be focusing on how do we simulate. It's very straightforward, and oftentimes, so our models, which is something I've alluded to, but haven't explicitly mentioned, are highly nonlinear. So to do analysis, it's very difficult to do nonlinear dynamic analysis. So we often linearize our system. And that ends up meaning that whatever results we have are local to this specific state. We don't know how far away they're from our particular state they're meaningful. And it's a little bit more difficult to extract these kinds of deeper understanding of our model behavior. However, with simulation, simulation inherently accounts for the nonlinearities. And so it's a very straightforward way to get a comprehensive picture of your system, as long as you're simulating enough and you have the tools to understand what those simulations are telling you. So that's going to be the topic for today. So these kinds of models, there are even more generalized ways to formulate them, but we typically will put into play a number of assumptions that make our lives easier and don't seem to really impact the relevance of the model to our case studies. So the first is a continuous assumption, meaning that we're not modeling any molecules discretely. We instead treat things as a continuous medium. And even beyond this, we don't typically model any kind of spatial gradients. We will often or almost always model our system at a constant volume. So even though a cell is constantly growing, for example, we don't model that increase in volume. We just assume that the volume is constant. We ignore, electrodiffusion and any kind of physical factors on the cell that, for example, might change transport equilibrium constants and otherwise make our lives difficult in a way that isn't relevant to the kinds of questions that we're asking of the model. And yeah, we just model that it's homogeneous and constant temperature. So of course, if your question is specifically pertaining to one of these aspects, like let's say you wanted to understand how the dilution of metabolite pools due to volume is responded to by your metabolic network, you're going to have to reintroduce some of these, or remove some of these assumptions and somehow explicitly represent the the factors that are being assumed away, but it very quickly gets very difficult to model. So these are very convenient to make these kinds of assumptions. Okay. All right, so that's the dynamic mass balance. So let's start to apply it to model reactions, and which is really the topic of chemical kinetics, and the basis for modeling kinetics, which is the mass action principle. So mass action, which I'm sure everyone has heard of in the context of a basic chemistry course, simply states that the rate that a reaction happens, which is the basis for chemical kinetics, right? We're talking about rates, is proportional to the collision frequency. So in other words, the more often two molecules see each other, the more they react in a proportional manner. So that's very intuitive that it would work like that, but that's the basis for our what we call our rate law, which tells us how the reaction rate V depends on the metabolite concentrations X. So for a linear reaction, let's just say it's a decomposition, then you have some rate, and that rate is going to be proportional to your concentration X. And so we define this proportionality constant that we call the rate constant. For a bilinear reaction, the collision frequency is going to be dependent on both of these metabolites, and so it's the concentrations of the two multiplied by each other. So we end up with these kinds of simple mass action principles, but this extends beyond simple metabolites to the protein level. Even here with a protein complex formation, you can model the reaction the formation of this complex from its individual subunits, and everything is still applicable. You could see here in the case of dimerization of this complex, formation of this, where you have two subunits that combine to form a single dimer. So this is the principle for all of the rate laws we're going to be talking about today. There are some specialties that modify this, special cases where the reaction rate isn't exactly proportional to the collision frequency, but I think we'll talk about that right here. Okay. So what does this mean for, it makes sense that this principle of mass action exists, and for small molecules hitting each other and reacting, it's very intuitive how this happens. They just see each other, and then if you've taken organic chemistry, you have any of these substitution, elimination type mechanisms that govern the kinetics. But what does this mean for enzymes? So biological reactions are typically a reaction between a substrate and enzyme, or an enzyme and two different substrates. So what do reactions look like for that? So here you're in the center, you're seeing kind of a schematic of how this occurs. You might have two different metabolites, and then two different binding sites in a protein, in an enzyme, that have to somehow dock these metabolites. So A has to find its site with the correct orientation, and same with B. And depending on an angle of collision, you might not have a reaction, and it has to be exactly situated. So the point is that every time these molecules encounter each other, they have a chance of reacting. And so the more of these metabolites there are, or complexes there are, the greater a chance that they react. But that doesn't mean that every time they see each other, they're going to react, right? So that's where the proportionality comes from. So there's some relationship between the rate and the concentration, but it's not like every single time they encounter each other, they'll react. And there are, you could think of it as if there are chances that, if there are factors that restrict the, what we call restrict geometry, or restrict the likelihood of them encountering each other with the right orientation, then we even can have to modify the terms on the mass action. So this is for restricted geometry cases. And these can either enhance or deter the likelihood of a reaction occurring. So so these numerical values of the not only the rate constants, but even the proportionality of mass action are determined by the frequency of a right collision and the geometry of the system within your cell. And so, you know, modeling all of these factors can get quite complicated, but typically we just ignore this kind of exponent that can pop up in the mass action coefficients, and just focus on the more basic mass rate law here with V equals Kx. The second point to note here is that K is a biologically determined variable. What does that mean? So in addition to K depending on collision frequency, orientation, the charge state of the protein, or I guess the charge state is a genetic parameter, depending on the temperature that the reaction is run, for example. It's really dependent on what this protein actually is. So you can imagine if the metabolite was negative and this whole area was filled with positive amino acid residues, it's actually going to make it harder for the metabolite to find its binding site. It's going to make it a rarer event because there'd be electrostatic repulsion. So the sequence of the enzyme actually impacts these rate constants. So for chemical kinetics without enzymes, you know, it's just dependent on the metabolite properties and the chemical properties of your system. For biology, we're really talking about Ks being genetic variables. So you can have good and bad enzymes for catalyzing particular reactions, and that ends up changing your K. And that's really a big part of kinetic modeling, is figuring out what the rate constants are for your particular organism, for your particular enzyme, because it's going to be changing depending on what the sequence is. So how this actually works for real systems. So before we were talking about modeling mass action with a single direction, just thinking about X. In real reactions, we have we can't assume that our product is a negligible concentration. All of our metabolites in a network are there because they're parts of pathways. And so we have a forward and reverse reaction, V plus and V minus. Each one has a conversion that it catalyzes, symbolized by this kind of double arrow here. And so we have a net forward rate and a net reverse rate with different rate constants, and they're dependent on different concentrations. And so we can define our net V, which is the production minus consumption, like a little mini mass action network, and get our full mass action rate law. And now we have to bring into play thermodynamics. So we have a production and a consumption. And so when these are perfectly balanced, our reaction is at equilibrium. And we can define an equilibrium constant, which is what is the equilibrium concentration, equilibrium ratio of the product over the reactant. And from a simple mass action, for a simple mass action reaction, you can see that this is equal to just the ratio of the rate constants. And so, again, this is very simple to think about. For a chemical reaction, once you bring enzymes into play, it gets a little bit more complicated, but we'll talk about that. And then for an actual enzyme, now we're looking at the Michaelis-Menten mechanism. So we're bringing an enzyme into play. Instead of just X1 to X2, we now have our substrate with our enzyme that is reversibly binding to form a complex. So that complex we showed in the last slide. And then irreversibly reacts to form a product and an unbound enzyme again. And we can get we can derive, and I'll show how in a second, an overall rate law for this reaction. So now we're not talking about individual V1, V2, or V minus 1 and V2s. This is now overall rate for S converting to P. And so now it's just in terms of those terms. But it gets more complicated, and now we have these Vmax and Km terms that everyone is familiar with. Okay. So there are different ways that we can apply assumptions in order to reduce the complexity of these reaction systems. So we can model these as network of mass action reactions, but oftentimes it's convenient for analysis and our understanding of how these reaction systems work to apply certain assumptions to collapse these enzyme reaction networks into a single overall rate law for the reaction, for the formation of our product. So here we're doing that for the Michaelis-Menten rate law by applying what's called a quasi-steady state assumption. I'll show what that is. And we can also apply a different assumption on called a quasi-equilibrium assumption, saying that this first binding step is at equilibrium, and get a very slightly different rate law here, where the term Km is a simple Kd. So our parameter interpretation has changed depending on how we've gone from our mass action network to an overall rate law. The other important concept is distance from equilibrium. So we talked about how for this kind of net reaction, we have an equilibrium constant, which is the ratio of the concentrations when your reaction is at equilibrium. But our reactions are not at equilibrium for the most part because we know that they're carrying flux, right? So oftentimes it's interesting to analyze how far they are from equilibrium, which is done as such. So we can define, so we've already defined our equilibrium ratio, which is the concentrations at equilibrium. We could define another parameter we call gamma, sometimes called Q, or reaction quotient. That is the ratio of the concentrations at steady state. So our steady state, we're assuming here, is a non-equilibrium steady state. And we can take a ratio of these two to say how far a reaction is from equilibrium. We can actually reformulate our mass action rate law in terms of a distance from equilibrium. So and we get something that looks like this. K1 times x1 is 1 minus gamma over KEQ. So this is just a reformulation of this rate law in a way that highlights the distance of the reaction from equilibrium. So when a reaction is at equilibrium, our gamma is equal to the KEQ, right? Because our steady state concentration is equal to the equilibrium concentration. And so this term becomes 1, and you have 1 minus 1, and our net flux goes to 0, which is our intuitive interpretation of what equilibrium means. So our forward and reverse flux are equal, and we get no net flux. Just to talk a little bit about thermodynamics, more about this equilibrium constant, where it comes from, and why we care about it. So you might be familiar with this framing, which is that there's this constraint that's derived from the first and second law of thermodynamics, that for a reaction to proceed in the forward direction, the Gibbs energy has to be negative, has to be less than 0. And if so, that this is a succinct formulation of that, is that whatever the sign of your V is, so if your V is positive, then delta G has to be negative. So V positive, delta G negative. Or if your V is negative, so if your reaction is going in the reverse direction, your delta G has to be positive. So in other words, your Gibbs energy of a reaction determines the direction that your reaction is going in. So what is a Gibbs energy? Well, let's think about how a reaction proceeds. So in a chemical kinetic perspective, we have, let's just think about this in an energetic perspective. Our reactants and our products have particular energies. This is a thermodynamic fact. They have a formation energy based on the internal energy of the metabolite. And so as your reaction proceeds from reactants to products, you have a change in energy, and that change can be positive like this, it can be negative, it doesn't really matter. But to proceed from positive to, or from your reactants to your products, you have to proceed through a typically higher energy transition state. So a state that those metabolites don't like to be in, and so it's a higher energy state, meaning it's going to be less frequently observed. When we're talking about kinetics, we're really talking about how often reactions can proceed over this curve. And so the lower an activation energy, the lower this energy increase is, the faster reactions can happen. With thermodynamics, the only thing we're talking about is what is the difference between the energy of the reactants and the product. So it's like an inherent favorability of a reaction. And that's really what goes into this KEQ term. So what goes into the Gibbs energy in the previous slide, in the second law of constraints that determines our reaction directionality? Well, it's two terms. The first is what we call a standard Gibbs energy, and the second is our reaction quotient, what we were calling gamma before, and on this slide is Q, our reaction quotient. So our standard Gibbs energy is just a simple function of our KEQ. A KEQ, which we already defined as the ratio of the metabolites at equilibrium, which is a metric of that inherent favorability of a reaction. So if you have a really large KEQ, it's easier to form product than it is reactant. That reaction inherently can run very well forward. And then our Q is that mass action ratio then, which is our actual steady state concentrations. And so we can formulate this just by saying that you can combine the natural logarithms there like this, which is that it's RT ln Q over KEQ. So it's that same ratio that we're showing on the other slide. So in other words, the inherent favorability, which direction the reaction goes, can be calculated by just comparing our concentrations at steady state to our inherent favorability in our KEQ. Okay. So just an example for a simple AB reaction, we have our constraint here, or our equation here, that we can then look at if this is positive or negative and determine the direction the reaction is going in. And we can calculate our mass action ratio, which is going to be B over A for this very simple reaction, products over reactants. And we can maybe assume, just for argument's sake, for illustrative purposes, that our KEQ is 2. So very, very slightly forward favorable. But that KEQ alone doesn't tell us what direction reaction is going in. We have to compare it to the steady state concentrations. So we can look at our equilibrium ratio and see that the products over the reactants are equal to 2 at equilibrium. So in other words, B equals 2A when the reaction is equilibrium. However, when our product is less than 2A, we know that our reaction is going to be proceeding in the forward direction, and when B is greater than 2A, our reaction is going to be proceeding in the negative direction. So all we have to do to determine what direction our reaction is proceeding in is look at our metabolite concentrations and compare them to our equilibrium constants for those reactions. As I already showed on a previous slide, thermodynamics and kinetics are themselves connected. It doesn't just tell us what direction the reaction is going, it does tell us something about our kinetic rate constants, and that's just shown here. So once again, we can look at our equilibrium ratio here. KEQ equals the concentrations, the ratio of the concentrations. But now we're bringing in this mass action rate law, which brings in the proportionality constants, the rate constants here. So we have our forward flux and our reverse flux with forward and reverse rate constants. And at equilibrium, we know that our forward rate is equal to the reverse rate. That's the definition of equilibrium. And we can substitute in our rate laws here. So we get an expression in terms of our rate constants. And now we can see that our KEQ is going to tell us something. It's going to tell us the ratio of our forward rate constant to our reverse rate constant. And so the thermodynamics, the KEQs are fundamentally tied to the kinetics. And again, this is for a simple reaction system, just an AAB. For enzyme kinetics, when we're looking at overall reaction KEQs that involve multiple sub-reactions, it gets more complicated, but that's something that we can handle. So this is just a preview of how that works. So in enzyme systems, we have a series of reactions, as we already saw for the Michaelis-Menten. We had, E plus S goes to X goes to E plus P. Here we have A goes to B goes to C. But our overall KEQ of A to C is just the multiplication of the individual KEQs. So you have a KEQ for reaction 1, KEQ for reaction 2, and the overall KEQ is just the multiplication of these. And this is just derived from the first law of thermodynamics that says the energy has to be conserved. And so we can use this to get an expression of our overall KEQ, which is this KEQ from A to C, in terms of the individual rate constants by subbing in these relationships we just derived. And this is called the Haldane relationship, and this is very useful. We use this to relate our overall KEQs to individual rate constants to help constrain, to help determine what these values are for kinetic modeling. KEQ is not an absolute term. It's the inherent favorability of a reaction, and some things make reactions more difficult to run. So KEQs are dependent on the conditions that we're running it on. Here's, for example, they depend on the pH because certain reactions or certain metabolites have proton binding groups. And so if you add more protons to your system, that makes these reactions harder to run. Here's an example for ATP reacting with pyruvate, which ATP, any phosphate containing group has a lot of protonation sites. And you can see that the reaction energy here, the standard Gibbs energy, which we said relates to our KEQ, has a very strong dependence on the pH. And so the reaction favorability, inherent favorability, goes down at higher pH and is more favorable at low pH. The reverse. This is the Gibbs energy, so it's more favorable at high pH and less favorable at low pH. So lower energy is easier to run in the forward direction. Ion concentrations, temperature, all of these things can affect your KEQs. And that especially matters for transport kinetics, where you have protons that are co-transported, and then you have to account for membrane potential and the charge of these metabolites. And so you have to have different binding equilibrium equations for all of these states, accounting for their charge, and as they get affected by the membrane, the typically slightly more negative inside of the cell. So it can get quite complicated to handle all these factors, but there are equations to do that. And so this has been kind of formally handled and typically gets accounted for by different software tools to estimate these constants. So just a little bit on where these constants come from. So typically you will measure KEQ by watching reactions to proceed to equilibrium and then measuring the ratio that those reactions or those metabolites ended up at equilibrium. And so you could say, okay, I measured the KEQ of this reaction. But for reactions that you don't measure, you have to estimate them somehow. So the majority of reactions still haven't been measured, what their KEQ is. And so oftentimes you can build these reaction cycles, for example. So as long as you build a complete reaction cycle such that there's only one unknown, everything else was measured, and everything is kind of connected, then you can use the first law, energy conservation, to guess what that last remaining piece is. That's one way. You can also do what's called group contribution, which is break metabolites into subgroups. So for this particular metabolite, alanine, you could say it has an amine, it has a carboxyl group, and then it has a methyl group. And each of these have some inherent contribution to the energy of the metabolite overall. And you can infer that with something as simple as regression by fitting what these energies are for your known data, and then applying these energies to reassemble estimates of the energies of new molecules. So it's kind of breaking your molecules down, figuring out what their components energies are, and then using this to estimate new metabolites that you didn't have good estimates for. So this kind of group contribution theory has a number of work, and our lab has published some of that as well. Okay, so with the time remaining, let's get back to kinetics and talk about how we actually can build models, or how do we actually understand the properties of kinetic models. So one of the main features of biology is that it's multi-scale. You have different scales in size, you have different scales in speed, and you have different scales in absolute number. And I'd say that both of these, size matters less for kinetic models, other than when we're analyzing some of our assumptions around diffusion and things like that. But the how fast and how many's are really what matters. And one thing to notice is that these things, these processes and metabolites span orders of magnitude, which means that there's going to, it's not, a dynamic network is not a bunch of kind of equal things interacting. You have really massive pools of metabolites interacting with really small pools or really small amounts of enzyme. And you have really fast and really slow enzymes all interacting together. So there's going to be a lot of separation in the time scales that things happen at. And that's really a central feature of dynamic modeling, is understanding how these time scales actually result in the overall behavior of your network. So for example, this results in a number of simplifications that we can do to go from very large networks of mass action systems to a simplified understanding of the dynamics of our network. And so I'll go into these each individually, but these are 4 of these concepts. This is not an exhaustive list, but it's 4 examples of how we can extract dynamic intuition for the behavior of our network. So these are time constants, variable aggregation, transition analysis, and then graphical visualization. So first, time constants. So time constants are just a measure of the time it takes to get a significant, what we call a significant change. So if you observe some kind of dynamic process, oftentimes this is, we learn this in the context of decay. So we learn half-lives. That's an example of a time constant, but any kind of dynamic process you can define a time constant for. You can also define an inherent expected time constant through analysis, which is kind of what's shown here. But a time constant is relates to some kind of dynamic change. So how much change has occurred, to how long to the rate that it was occurring. So the slope here, which gives you an estimate of how long it took to occur. So what's nice about this is this can be inferred very generally. So oftentimes you're simulating networks and you don't know what kind of time window to look at. This can tell you inherent time constants for different processes within your network. So for example, if you have some kind of pool and it has a production and consumption reactions, you can define a time constant as that pool size P over the what we call the turnover rate. So how fast this pool is getting changed. This can be extremely relevant in biology when you think about the fact that I just mentioned that there's orders of magnitude differences in pool sizes and rates. So sometimes you will have a very large pool being acted on by very small fluxes, which creates a very long time scale. It takes a very long time for very small fluxes to change a large pool. And what that means is we can often neglect this process and just focus on faster processes, or vice versa. We might be interested in the very slowest aspects of our network. It allows us to separate the processes in our network based on the time that they're occurring on. This kind of separation can result in a simplification in our network due to through aggregation of our variables. So if we look at a network like glycolysis, we're going to have fast and slow processes within even a small network like this. And so if you have a very fast process, you end up with what's called pooling, meaning that any kind of dynamics that were going to happen very quickly. So if we have our full network at this first layer, this highest layer, whatever the fastest processes that are occurring are going to happen immediately. So at the fastest time scale, let's say 10 to the 9 seconds as an example. And then they're going to relax. They're not going to move anymore on slower time scales because everything that was going to happen already happened. And then they become kind of internally relaxed. So they become an aggregate variable. And so as you go along longer and longer times, you get this kind of pool aggregation. And so that ends up kind of simplifying your network. And you can get fast and slow subpools, and you can analyze those. You can also analyze state transitions. So we can think of either, we're either at steady state, which is where we start, or we might move to a new steady state, and then we can analyze the transition between it in different ways. So we could analyze the properties of this transition period based on whether it exhibits certain kinds of oscillations, whether it overshoots, whether it has kind of a damped, smooth landing. So this is another way we can do analysis. And then we can also do different kinds of graphical visualizations, either by mapping dynamics onto a pathway and examining where things are occurring. We can look at time profiles of individual elements, or we can look at dynamic phase profiles, meaning we can plot one metabolite or complex versus another and analyze this over time. So let's get into that last piece a little more. With it, we have about 10 minutes left. So I was talking about how you can plot this. And let's talk a little bit about how you actually simulate networks to obtain some kind of understanding. So here we have a very simple reaction here, which is some kind of perturbation of the increase in an ATP load. So we have a reaction that consumes ATP, and we can just we have to do some kind of perturbation around away from steady state. So in this case, we're perturbing the rate constant K, increasing it by 50%. And so we simulate it over time, and what do we see? So one thing that I mentioned repeatedly already is that dynamics happen at multiple timescales. And so oftentimes it's illustrative to make different plots at different timescales. And that's what's shown here. So we have a fast, intermediate, and slow scale on our x-axis. And so our fast dynamics, you can see, there's not really very much happening. But once you start plotting at the hours level, you start to see different dynamics. And then at the days level, you even see different dynamics once again. So you can see that there's a very slow increase of glucose, for example, over time. So oftentimes it makes sense to find dynamics that are relevant to your system or to your questions and plot them in that time window. We can also, as I mentioned, plot one variable against another, and you might ask, which variables do I pick? What is meaningful? Well, oftentimes you don't know, and so you want to just create one of these, which is called a tiled face portrait, where you take all of your variables and you plot them against each other to see what kind of interesting interactions can show up. So what is considered an interesting interaction? Well, you can see motions that occur on a line like this, meaning that they're moving proportionally. This can actually identify variables that are pooling, variables that are moving together and have formed an aggregate pool. So you can look for correlations even in these plots to identify that numerically. You can find where more interesting kind of dynamics are showing up, and then do a deep dive into what kind of transitions are occurring over time there. And you can find completely independent variables that don't seem to have any relationship to each other. So this gives you a quick snapshot of the interaction between variables. You're no longer just looking at one variable at a time. You could even imagine clustering this based on shape to find groups of related metabolites, that type of thing. And then, yeah, this is just more on the dynamic behavior here. You can identify, you can create these phase portraits and plot them. Here, really nicely labeled is the different times that were occurring. So you can see a very fast regime and then go into a slow regime where things have, and then you can identify the time scales at which most of the dynamics have occurred. So here you can see there's a big jump that occurs between 0 and 0.1 seconds. And if that's something of interest, you can take a look at that. So just a lot of this is the same kind of material here, but just to reiterate, if you have this kind of low dynamic dimension, that can mean this kind of straight line showing up. That can mean that these variables are related and have formed a dynamic pool. And you can then say, okay, these are maybe all one dynamic variable, and I could simplify them and get a simpler representation of the system, whereas this is a more complex response that seems to have dynamics all the way through the simulation and might be one of the drivers of the system. So these kinds of plots are helpful for that purpose, and you can do the same thing with concentrations, more or less. Okay, so just to hit a few more dynamic visualizations before we end. So we already covered time profiles. So you can have you can look at single variables or two related variables, and you can do plots on multiple time axes. You can also do them all on the same plot, so showing 3 different time scales all on the same thing. This kind of inherently happens whenever you do a log scale. These are the dynamic phase portraits that I mentioned, where you have an initial time, and then you can just watch it evolve to its infinite time, and you can get these kinds of relationships that you derive from it. So either conservation, meaning that if you're looking at 2 pools and they have this kind of trade-off, that could mean that they're part of a single pool. So if you add to one, you have to subtract from another. You can see equilibrium relationships show up, which is one example of pooling. You can see dynamic independence, and then you can see kind of this more interesting perturbation. If you see circular motion exactly, that means you have a perturbation that returns to the same steady state. Yeah, sorry, this is all material I've gone in, so I think I can probably skip ahead. Okay, so now let's talk a little bit about building this up to the network level. So I already mentioned how you can build mass action reactions for individual reactions. So as we do this more and more, we end up with our stoichiometric matrix, which is something that we talked about on the first short course session about a month ago. We can also derive a number of properties of our network from this that are probably most useful for network analysis as opposed to dynamic modeling. But there are some variables we can create that represent the pools, these dynamic pools that we can determine. And these can be either intuitively built, or determined after the fact and used to create aggregate variables that you then plot for different analyses. So you might, for example, model all of glycolysis or track the phosphates in glycolysis, or you might track certain different energy pools or cofactor pools and plot those as a single aggregate variable instead of separately. We already touched on node maps. We can plot the dynamics around individual nodes to show dynamics in production and consumption. And we can also do pathway analysis. And this is derived from the S matrix, but isn't terribly important here. What is important is to talk about our steady state. So we always have to define some initial condition for our dynamic model. And we often will pick a steady state. So a typical workflow will involve flux balance analysis, actually, where we will first model our system at steady state, and then use that as a reference point to start to do dynamic perturbations. So I already mentioned that a steady state doesn't move, right, dynamically. So you can't use a steady state as an initial condition, but you can define a steady state to reference point and then perturb away from that to model a dynamic, some kind of transient perturbation, whether it's engineered or representing some biological process. And so this is just an example of what this typically looks like for glycolysis. You have your upper glycolysis that has some flux, and then that doubles for lower glycolysis approximately because you have this split. And then you have some boundary conditions that you have to define also that make your network an open system. And that's what enables it to carry flux at steady state. And similarly, you have to define steady state concentrations. And typically these come from metabolomics data along with estimates. So all of this you have to define ahead of time to start to parameterize your kinetic model. And then once again, you'll be plotting deviations away from this over time with the kinetic model. If you have these, then you can define what's called a pseudo-elementary rate constant. So this is in cases where you do not have explicit kinetics determined for your enzyme. So for most of the enzymes in the case studies we'll talk about, we have these Michaelis-Menten-like rate laws derived to determine the kinetic constants. If you don't have that, you can derive an approximate model rate law for your system with mass action, just by defining a mass action reaction for your overall reaction and subbing in those steady state values I was talking about. You can sub in your concentration, or your flux, your metabolite concentrations, and your KEQ, solve for this last variable, and you end up with kind of an approximate reaction. And we end up doing this oftentimes for boundary reactions or for reactions that we don't know the kinetics of or want to approximate and don't want to do a detailed model of. This is a good placeholder rate law, and it gives a approximate local approximation of your kinetics. Okay, so leading into this exercise, let's talk about what we're going to be doing. So first, we will be setting up our model. So setting up our dynamic mass balance system. What does this mean? It means we have to define our network. We have to define, just like with our constraint-based models, we take the entire metabolic network, but with the dynamic model, we're typically not modeling at genome scale. We're going to model a pathway or several pathways like a small network. And so we're going to extract some kind of network out of our stoichiometric matrix, out of our broader global model. And then we're going to have to define some kind of form for our rate laws. And now this can be those simple mass action perks, or it can be a detailed description of the enzyme mechanisms. And then we end up with our dynamic mass balance system that is dependent on some kind of concentrations and rate constants. Then we have to define what those parameters are, as well as the initial conditions for our simulation. And this is where the steady state comes in handy. as well as any kind of parameter fitting we've done so far. These rate constants often will take into account thermodynamics as well. It really depends on how you set up your model. And then once you have this, you essentially have a differential equation system, and you can model that with any kind of ODE solver. And in this case, that's MassPy that is doing that, and it talks to different solvers like libRoadrunner, other packages that do the solving. And then when you have your solution for X over time, you can then do all this graphing, plotting, analysis that I was just showing. So how are we going to do this for this exercise? We're going to be using MassPy, which is the tool that Zach Heyman developed. This is a CobraPy compatible tool. So it was built on top of the package that we introduced for flux balance analysis, CobraPy, and uses a lot of the same namespaces when dealing with the models. So that's good to be aware of. It's intended to be compatible with CobraPy workflows. So you can go from constraint-based modeling to kinetic modeling of your same network. And that is indeed what we do in this workflow. We'll do both. So it handles the constraint-based modeling, some thermodynamics, it does dynamic simulation, it does some dynamic analysis, which we're not going to go into too much today, visualization, and then handling enzyme mechanisms, which is the topic of the second lecture for this session. So it's a comprehensive kinetic modeling package for building and analyzing these models. So this is the PyPy location of it. It also has a public GitHub as well as documentation, and is used in the textbook that I showed earlier, Systems Biology Simulation of Dynamic Network States. So a lot of resources built using this. So plenty to check out if you want to learn more. We'll be covering this. Just an overview of what you're going to be going through. The first is setting up the equations, so actually importing and building the reactions metabolite by metabolite, and then specifying these all the necessary parameters, your initial conditions, and your parameters, your rate constants and equilibrium constants, and then simulating it. So this is solving that differential equation system, and then plotting. Okay, and this is just an example of the output. So whenever you do a simulation, you end up with some kind of table of numbers, and it comes down to these plots, and you have to somehow extract that. Okay, extract some information, and Zach will go over how to do that. So now we're going to be stepping through. If you want to follow through this is going to be in GDGA short course, module kinetics. Part 1 exercise, MassPy tutorial. So that's what we're going to be following through. I would suggest maybe we take a short break, maybe 3-minute break, come back at 8:10, and then start. How does that sound, Zach? I'll pause the recording and stop sharing.
Works for me.
Okay. All right, thanks everyone. So I'm going to stop share and pause.
Is my screen being shared?
Yep, looks great.
All right, cool. So, are we ready? All right. So, this is just a basic MassPy tutorial. For those of you who want to follow along, this is, again, part 1 exercise. This is also available in the MassPy documentation in case you want to go back and look at it in additional detail later. Now, what we're going to do in this notebook or this exercise is we're going to build this network right here. And just for the sake of convenience, we have all of the reaction rates specified right here. We note that we have a fixed input, B1, for the AMP coming into the system. We have our concentrations and our parameters. And now this works for the smaller system. We have all these values. But often we don't always have these values, and so we have to solve for them. And so what we're going to do first is we're going to build just this small network inside of this. We use the form and distribution reactions. And so to do this, let me restart my notebook. we start by importing MassPy and just some basic classes, specifically the mass metabolite, the mass model, the mass reaction. And these inherit from the CobraPy package and behave very similarly. They're just prefixed with mass, so you're able to tell the difference between the MassPy and the CobraPy objects. And the first step we do is we create a model. And it's pretty simple to create a model, it just needs an identifier. And the system we're going to model is, again, this phosphate trafficking system, specifically around the ATP, ADP, and AMP metabolites. And so when you first create a model, it's initially empty, no metabolites, no initial conditions, no reactions. And so what we have to do is then define those metabolites. And MassPy is optimized mostly for the big IDs. However, you can provide any kind of identifier you want, as long as it's SBML compliant. And so we can go ahead and we can form the metabolites by simply providing an identifier. We can also provide additional information, such as the name, the chemical formula, the charge of the metabolite, and the compartment in which it resides. And we do that for these three metabolites because they're the first three that we're looking at for the smaller system. And once we've defined those metabolites, we are able to set their initial condition values. And so to set the initial conditions, essentially, you just use this initial condition attribute. Metabolite.initial condition equals the value. And there's also these alias methods, .ic. So if you get tired of typing initial condition, you can use IC. A little bit less important now that Jupyter notebooks have all these tab completion methods, but still, once you've defined the metabolites, we can go ahead and print out those initial conditions and check the values, make sure we define them correctly. And then we can go ahead and look at the ordinary differential equations the metabolite is a part of. And because we have not defined any reactions, we get nothing returned, none. And so that leads us into our next step, which is defining the reactions that these metabolites participate in. And so just like the mass metabolite object, we need to provide a unique identifier for the mass reaction object. We can also provide a name. And then very importantly, we provide whether this reaction is reversible. And this has a different connotation in MassPy, where reversible does not refer to the flux bounds, but rather reversible refers to the kinetic reversibility of the reaction. And this essentially is what determines whether or not a reverse rate component is generated for the rate law. Most all reactions kinetically are going to be reversible, but sometimes we do have a need to define them as irreversible, like in this smaller system here. So we go ahead and we can define the reactions. And as you can see, if we wanted to, we get this nice little table in Jupyter notebooks where we can see reaction identifier, its name, address. But as you can see, no metabolites have been added to this reaction. So we're going to go ahead and we're going to add the metabolites, and it's very similar to how CobraPy adds metabolites. In this case, we have reactants are negative coefficients and products are positive coefficients. And we simply take the object that we built earlier and we provide it as the key, the coefficient as the value, and we can form these metabolites. And now we can see what our reaction looks like. If we look at the form reaction again, you can see it's been filled in, and the second one gives us our reaction in terms of the names, which is why it's nice to define a name for the metabolite. Now, once we've defined the reactions, we can go ahead and we can create their parameters and define those. And so just scrolling back to the top of the notebook, you can see that we have all of our parameter values defined up here. So we're just going to take those and define them down here. And the way we do this is for a forward rate constant, it's just the forward rate constant attribute. For an equilibrium constant, again, just the equilibrium constant attribute. When we define the parameters, we get, they all get stored in this dictionary called parameters, essentially. And you can see that the identifiers for these parameters are automatically generated, and they're based off of the reaction identifier. KF for K forward, Qcqa for equilibrium, and then their values. And then we're going to do this again for the forward rate constant of the use and the form reactions. We also have this alias method for forward rate constants, Kf, as well as one for equilibrium, Keq. And so when we run that, we get these values. And finally, we can take these reactions and we can add these reactions to the model. And now we can see that our model now knows it has 3 metabolites, 3 initial conditions, which we have previously defined with the metabolites, and 3 reactions. And note that whenever the reactions are added to the model, any metabolites that have not been added to the model will be added automatically. And now that we have a model with reactions and metabolites in it, we can look at the stoichiometric matrix. And as you can see, we get this NumPy array where the stoichiometry is defined, but sometimes we wish we could see the labels and know what's there, what isn't, and which row corresponds to which metabolite, which column corresponds to which reaction. And that's why we have this method update S, which is essentially update stoichiometry. You can use it to generate a stoichiometric matrix, and you can use it to essentially update the type of array returned. And this includes NumPy dense matrices, which is what we see here, some SciPy sparse matrices, and a data frame, pandas data frame. You can also change the D type. And in this case, since we're working with integers, we're just going to set it as integers. We set update model to be true, reflecting that we want our model to remember this array type and this D type every time we call up model.s. And when we run it, we get the stoichiometric matrix, but this time with row and column labels. So now it's a little bit easier to tell what the stoichiometry of the model is. And once we have this matrix generated in the model, we can also start looking at the reaction rates in the metabolite ordinary differential equations. And these are generated through the SymPy package underneath the MassPy hood. But essentially, it's putting together all of the rates and ordinary differential equations automatically. And so we can print that out by just doing reaction.rate. And we can see here that we get the distribution reaction. We have the squared term because we have 2 ADP as the reactants, and all over the equilibrium constant. And this is the mass action rate for it. And once we have the mass action rates, we can also look at the ordinary differential equations of the associated metabolites. And no longer is it a none value, but instead it's taken all of the rates and combined them to form the dynamic mass balance for ATP. Now, once we've defined those metabolites and those reactions, we can look at some of the properties of the stoichiometric matrix. One such property is the null space, which essentially gives us the pathways through the system. And so we run this, you can see that we get a pathway, one particular pathway, because this is a very small model, just the 3 reactions in it. And the pathway is essentially the cycle between the use and the formation reactions. Similarly, we can run the left null space function, which gives us the conserved moieties, or the conserved metabolites in the in the model. And these are time invariant properties. And so when you have a closed system like we do, in this case, the time conserved, the time invariant metabolites are based on the total amount of ADP, ATP, and AMP, essentially the total adenosine phosphate metabolites in the model. And there's one for each in this pool. And that is essentially a pool that, if we plotted, would be constant. Now, the reason we haven't, going back again, just up to here again, we have this model. We've built the use, the form, and the distribute. So it's a small closed system model. And more often than not, we actually are going to be taking models and we're going to be that exist and we're going to be expanding them. So we're going to take this existing model that we just built and expand it with the buffer reaction, right? For for ATP, ADP. And we're going to also add in a sink and a source for AMP to make this an open system. And so coming back to here, we take this model, and again, we pretty much define the metabolite form the same way we did before. This time we give it the identifier B for buffer and BP for buffer with phosphate loaded. And we define a reaction for the buffer. And we can add those to the model. And it's important to note that because this is object-oriented programming, essentially, we need to use the metabolites that already exist in the model when we're adding new metabolites to the reaction. And so we're going to take this buffer, we're going to add it as a reactant along with ATP that exists in the model. And the get by ID is just an easy method to get metabolite from the model using its identifier. We're going to do the same for the ADP as a product and the buffer loaded as a product. And this reaction is essentially representing, we print it out, reaction is essentially representing the transfer of a phosphate from ATP onto this buffer molecule and releasing an ADP in the process. So it's essentially storing the phosphate to be used at a later point or in some other larger system. Now, we go ahead and we set the parameters for the buffer reaction, which were again provided for us in the diagram. And this time, instead of setting them directly on the reactions, because our reaction already exists in the model, we can simply use the model.update parameters method. And update parameters is convenient because very often we want to try different sets of parameters on these models and change multiple parameters at once and in order to create a new model. And so this provides an easy way of updating the parameters rather than having to go back and reset them all for each individual reaction. Now, something important to note is update parameters requires knowing the parameter identifier for each particular parameter you want to update. And so reactions have these methods built into them if you aren't sure what the identifier is, where called KF string, KEQ string, and that essentially produces the identifier of the parameter. In this case, K buffer and KEQ buffer. And when we add the buffer model, the reaction to the model and update these parameters, we can see that our left null space expands, which means we have another conserved time invariant in the system. And this time it correlates or corresponds to the total amount of buffer in the system, because the buffer is no longer the buffer is not entering or exiting the system, it just remains in the system. And that's what this second pool down here is telling us. While we still have not put in any we have still not opened the system up, so we still have a conserved amount, or the conserved ATP, ADP, and AMP metabolites in the system. Now, from here, we need to determine what the parameters are for the buffer reaction, specifically, we need to determine the initial conditions for the buffer reaction. And we were told the diagram that we have a total amount of buffer is equal to 10, but we need to set the individual values for the initial conditions, the initial value for just the buffer and the loaded buffer. And so to do this, we use SymPy to solve the equation symbolically. And we can import a couple of functions to help out. And we also import this method strip time from MassPy in order to take the time dependencies away from the rate equations. And so the first thing we're going to do is we're going to define the buffer equation symbolically, and have to import it first. Symbolically define the equation, and pprint is just a SymPy method for pretty printing the equation. In Jupyter notebooks, you just call the equation, it'll try and do it using a latex printer. This sometimes looks good, sometimes it doesn't, so be cautious when you're using it. Now, when you go from here, we can go ahead and we can take the rate equation and we can strip the time dependency from it. And now you can see these go from having little functions of time to the function of time has been stripped, and we just are left with the symbols. And this makes it easier to perform substitutions on the symbolic expressions. And so we go ahead, we strip the time dependencies, and we set it equal to 0 because this rate equation is at steady state. And we substitute in the initial concentrations that we do have in the parameter values that we already set. And so that's what this dot subs is doing, is it's essentially equation dot subs puts it into the system, or puts numerical values in for the variables, and we get an equation. And then these equations can then be solved using the SymPy solve method to determine, okay, what the initial concentrations are for the buffers and the loaded buffer. And so we can still see that the total concentration is 10, but by solving the equations, we now know that the initial condition for the buffer molecule is 2, and the initial condition for the loaded buffer molecule is 8. And we can now update those initial conditions into the model. And we're going to go, so we can go ahead and we can iterate through. This is a dictionary it's produced. And so we can iterate through the symbols and through the solved values. And we use a copy of the dictionary here only because we're going to go ahead and pop the entry out of the dictionary for setting it. And so we get the metabolite from the model. And by using the get_by_id method, the symbolic representation of the buffers also happens to be its identifier, just BBP. So the get_by_id method works well for a lot of these cases. And we go ahead, we create a dictionary that's got the model metabolites for the buffer mapped to their solution values. And then we use that dictionary with an update initial conditions method to put those values into the model. And what we get is the model with now all of the initial conditions in it defined. Now, the next step is to expand this model to add the sink and the AMP source and the AMP drain reactions. And we can use this method, add boundary, that was defined in Cobra, to add these reactions. And to do that, we simply have to provide the metabolite object we want the boundary reaction to be added for, the type of boundary reaction, and we can provide a specific identifier for it if we want to follow our own conventions instead of using the exchange, the sink and demand prefixes. And so in this particular case, that's what we're going to do. And we can define these reactions. And you can see that the AMP drain and the AMP in, they're both demand reactions, because by the very nature of a demand reaction, it is essentially an irreversible reaction, either entering or exiting the system. And so when we create this boundary reaction, we get a metabolite that a pseudo-metabolite, or essentially a boundary condition that represents this null value. We can access that through this boundary metabolite attribute. And that allows us to set a boundary condition for the boundary metabolite using the add boundary conditions method for the model, and a dictionary where the string is the key and the value is the boundary condition value is the dictionary value. And from there, sometimes, again, we want to switch the direction of the reaction because it will be more intuitive, or in certain cases like this AMP in, we want to make sure that if it's irreversible into the system, not irreversible leaving the system. And so we have this method called reverse stoichiometry, and we do it, use the in place is true argument, essentially, to say we want to reverse this reaction in place. Otherwise, it will return a copy of the reaction. And by running this, you can see now that the stoichiometry has been switched, and we now have this boundary metabolite on the left, and then entering the system to become the AMP inside the system. And now, by having the addition of these boundaries reactions to the system, we actually have, in opening the system up, we've actually introduced a new pathway into the system. And so when we look at the null space again, we can see that pathway, which is essentially now the AMP coming into the system and AMP leaving the system. And note that we also still have our original pathway, the cycling between the use and the form reactions for ATP, ADP. And now lastly, sometimes we want to define a fixed input or some custom rate, maybe a Michaelis-Menten rate law rather than a mass action rate law. And so what we do is we have this method here called add custom rate. And as you can see right now, this is just a simple mass action rate law coming in. It's forward reaction based on the buffer value and the forward rate constant. but we would prefer to define a fixed input rate, as in our original diagram, using this variable B1. And so we have to define a custom rate by providing the reaction, the custom rate as a string. In this case, it's just B1. And then because B1 is not a parameter that currently exists in our model, we define it as a custom parameter, specifically with its initial value, which is, again, 0.03. And when we define this, the model's rates now include this custom rate. And whenever you call the model rates, everything else is still their mass action rates, but you have your fixed input for the AMP drain. And so now that we've defined the model, we want to go ahead and we want to ensure the model is complete before we can simulate it. And essentially,
quickly ask one question, Zach,
sorry.
About the add custom rate, you just added here a custom rate constant, but if you enter also the metabolite concentration IDs, this function will automatically notice this or
yes,
recognize this as the metabolite ID.
Yes, this function. So the way this method works I'm glad you brought that up. The way this method works is it can identify every parameter in metabolite that already exists within the model. So for example, AMP is in this model. So if we added, or let's use ATP, just for the argument's sake, we added ATP in here because it already exists. It's able to identify ATP and then put it into the rate. The issue only comes in if we were adding in something that does not already exist, like this parameter here, B1. So that in that case, we have to provide it as a custom parameter so it knows it exists. This is also why it's just in terms of creating a model or working with the model, it's better to start with the model, add the metabolites, add the reactions, and then go ahead and define the rates, because it will be able to then determine, okay, this already exists in the model and versus this does not. We need a new variable. So does that, any other questions about that? No.
Not from me. That sounds great. So you can actually use anything that's in the model. Create your own rate law, whatever that is, apart from mass action kinetics. That's good.
Exactly. And so it's primarily handy when you want to define a fixed input for exchange reactions or boundary reactions. It's also handy if you want to use a rate that's not a mass action rate, like a Michaelis-Menten rate law, or if you want to create, for example, some sort of reaction that has a complete like a time dependency in addition to the time dependencies from the metabolites. You can always put in a time variable like times t, kind of a deal. And so once that's done, we go ahead, we ensure the model is complete. We want to double check to make sure that our rates make sense and our ODEs make sense, especially if we were playing around with this. Since Jupyter notebooks don't run always in a top-down fashion, we can kind of jump and run cells as we want. We can just quickly inspect the rates, make sure, okay, yes, B1 is still our fixed input, and these rates are all kind of what we expect to see. You can see that these three rates have not changed at all since adding the buffer. However, you can see that there's a rate for the buffer now added to the model, as well as the drain and the input. We can look at the model ODEs and again, look at them. And before running a simulation, we would want to just do a quality check on the model to ensure that everything is there for simulation. And so there's this handy method called Qcqa Model, which will run a series of checks on the model based on the argument flags. And more often than not, the ones that we're interested in looking at are whether all the parameters are defined and all the concentrations are defined. And if you note that when I run this check, it says that this model is not simulatable. And the reason for that is because I forgot to define the forward rate constant for the AMP drain. And if we're missing a numerical value, we cannot simulate it. And so this report is handy because it essentially gives me a hint as to, okay, I need to go back and define this value. And so I can define it and run the check again. And now the model is simulatable. There is no issue with missing parameters, and we can go ahead and simulate it. So this is kind of the model construction side of things. And from here, what we're going to do is we're then going to go ahead and simulate this model. And just for the sake of this tutorial, I'm going to take this model, which we had constructed to be at steady state, and I'm going to update these initial conditions so they are not at steady state. And so we're going to go ahead, we're going to import a simulation object, update these initial conditions just so our model is not steady state for starting, again, for demonstration purposes. And so now what we're going to do is we're going to load this model into the simulation. And the simulation method is essentially designed to hold an ensemble of models as well as a single model. And so that's why you need to first define a reference model. What is the base model for the simulation? Since we're only working with one model, it's just our model. And if we set the verbose argument to true, then we get these little printed statements telling us whether things were successfully loaded or not. It's just a way of, if it wasn't successfully loaded, you would get an error message, but sometimes it's nice to have them printed out to the screen. And so we can go ahead, load this model into the simulation and save it as the simulation variable. And again, something very important to note is underneath the hood of the simulation is this Roadrunner object from the libRoadrunner package. And libRoadrunner is a simulator that's designed, is a very high performance simulator designed specifically for SBML compatible models. So it comes back to, we need to make sure that our identifiers are SBML compliant. More often than not, this means if we have an identifier that starts with a number, for example, we might have to add a little underscore, some sort of prefix to it, just so it's not, does not start with a number. Luckily, that is not an issue for any of the models we are dealing with in this particular notebook, but it will come up in later in the part 2 exercise. So we go ahead, we define the simulation, and when we look at the reference model in the simulation, we can see that it's the exact same model that we had just created in here. And we can also go ahead and access, if we are interested or prefer to use libRoadrunner for some reason, we can go ahead and access that object directly. This helps for any sort of debugging or using some of the methods that Roadrunner provides. And after we've loaded the model into the simulation, something important to note is that the model will actually extract, or the simulation will actually extract out the initial conditions and the parameters of the model. And so if we're interested in extracting those out, we can use the simulation.get model simulation values method for a particular model, and we can pull these values out. And as you can see, the values loaded into our simulation are reflective of those in our model. And this is important because if we run a simulation and want to change the numerical values, the reference model will always be updated. But the more models you add, the harder it will be to keep track of all of this. And so what we do when we have a ton of models is we just simply store just the value sets. And so once we have our model loaded into the simulation, and we've already verified from previous steps that this model can be simulated, we can go ahead and we can simulate the model. And that's easily done by using the model.simulate method, or sorry, the simulations simulate method. And the very basic input for the simulation method is essentially which model do you want to simulate? And in this case, our model that we've defined before, and a time vector. And if we just provide the input in, or starting and end times, then it will, libRoadrunner will go ahead and make the decision on how many time steps we need. And so we can always provide a number of points if we feel that we need more by adding in a third argument, but in this case, we won't need that. And so when we go ahead and simulate a model, we get our solutions, and they come out as 2 solution objects. The first one is for the concentration solutions associated with the model. The second one is for the flux solutions associated with the model. And so it's helpful to take the solutions and split them into their separate, the list of solutions, and split them into their separate objects. And so we're going to look at the concentration solutions. And these concentration solution objects are essentially just a fancy dictionary with an identifier. So using the dictionary.items method will work on these objects. And as you can see, we can print out the solution values in this loop. And we can, by default, the solution values are going to be NumPy arrays. However, often we want to use interpolating functions, so then we can change the time points or smooth out the simulation results if we get little artifacts or noise in there. And so we can set inside the simulation function, when we call simulate, we can set this flag for interpolation. Interpolate equals true. And when we run the method now, instead of getting NumPy arrays, we get SciPy interpolation functions for each object. Very handy for when you want to look at a specific time range, let's say like 50 to 70 in this particular time vector. And then something else to note is simulations can get complicated quickly, and sometimes we need to adjust the integrator options. So by calling sim.integrator, we can see what these options are currently set as. Right now, the integrator being used is the CVO integrator from a powerful package called Sundials. It is an industry gold standard, very quick for performing simulations. Definitely would recommend using this one in many cases. We can also set the relative and absolute tolerances, whether the system is expected to be stiff or not. And something to note is when we're working with enzymes, the system is going to generally be very stiff, or enzyme modules, or enzyme kinetics, the system is going to be stiff. So it's good to keep this flag as true. Maximum number of time steps, or maximum number of steps or iterations. We also have the maximum time step, minimum time step, and initial time step. A value of 0 means that the simulation algorithm will decide for you. Sometimes, though, it does not pick a value that you would like, and so you can set these values yourself. And then one that's important for us is variable step size, as we tend to simulate across multiple timescales. If this is set as false, then we end up with a linearly evenly spaced linear points. And these points may not capture the fast dynamics that occur if the time vector that's generated goes from 0 to 1. You miss everything that might happen on the 0.01, 0.001, those earlier timescales. And then lastly, maximum output rows. This is mostly a method to help prevent too many values from being put out all at once. And for smaller models, it's not usually a problem. For larger models, sometimes we need to change this value so we can get more solution points. It just, again, it depends on the model. So knowing these integrator options and how to manipulate them is very useful. And because these are libRoadrunner methods, we actually get this handy method involved called getDescription. And so if we call the simulator.integrator, and then use the roadrunner.getDescription method and provide one of these options, in this case, variable step size, you can get the description for it. And this description is a brief description. Usually it will be what you need to know. If not, it's also available in documentation. And then going back to the variable step size point, if we change this value, for example, our time points are now evenly spaced. We change it to true, they're now logarithmically spaced. And so it's an important flag to know about, depending on the type of simulation you're performing and what you're interested in looking at. If you're not interested in the short time scales, you still may want to set this as true so you don't have any of the dynamics of the short time scale skipped over and get some artificial results. So essentially, if you have any questions about how libRoadrunner works, again, the documentation is very clear as well. It performs a lot of MassPy takes care of a lot on for the user. But that being said, sometimes you want more advanced control, and that's where knowing the libRoadrunner package can be helpful. But now, just moving on from there, once we have a simulation result, we want to then go ahead and look at those solutions. And so again, when we take them, we're just going to take the model loaded in fresh for the simulation and generate these new solutions. And the mass solution essentially contains keys, which are, it's a dictionary. It contains keys that are the string identifiers of metabolites, like ATP, ADP, AMP in this case. And if we have interpolate set to false, we get a numerical array. And by default, it's set to false here. And so when we go ahead and we print out the first 10 solution values, just using simple list slice, you can see what they are. And we also have this unique ability with the solution objects to treat the solution keys as attribute accessors. So instead of doing the dictionary method of accessing, you could just do solution.metabolite, or if it's the flux solution, solution.reaction. Now again, this tab complete does not work if you do not have an SBML compliant identifier, which usually means if you're starting with a number or like a dash, essentially characters that may have other mathematical meaning to them. And so it's one of the big sticking points of using this type of simulator is you always want to make sure you're working with SBML compliant models. And so go ahead, we can print out the solution again for the numerical array. And sometimes, especially for plotting purposes, we want the associated time vector that was generated with this solution. And so the solution object keeps that stored, and it can be accessed using the .time attribute. And looking at just the time, time for the first 10 points again, you can get it. Now, more often than not too, we like to work with data frames and have it in a bit more of a clear method. So we have this solution.toFrame method, which will simply generate this data frame based on the solution stored for each metabolite, and put the time vector as the index. And from there, we can go ahead and we can start plotting. And there are multiple ways of plotting a solution. Sometimes we just want to quickly view what the solution is. And so we have this method built in to solution objects. It's called view time profile. And if you have matplotlib installed, then it will work. And so just by calling the method, we get a handy automated time profile generation. We get a handy generation of a time profile with something like the title, the legend, and it's usually in a log scale because of what we're looking at. If we want to change this, we can add the plot function argument. We can set this to, let's be semi-log x. We can see different set values. However, it definitely, we have a little bit of trouble differentiating some of the values that are very close to each other. So we're going to go back to looking at it in a log scale. And from there, we can see that when we simulate this model for a long enough time period, our model returns to a steady state as indicated by these flat lines. And our steady state is essentially representative of the initial values that we had computed before, with rounding errors included. So you can see this rounds to 4, this rounds to 16, this rounds to 0.1, this rounds to 2, and this rounds to 8. And so we can always round the variables. MassPy doesn't do the rounding automatically, only because depending on what scale you're working with, sometimes you don't want the values to round if you're intending for all of them to be like times 10 to the minus 6 or 10 to the minus 8. So just something to keep in mind. But what MassPy can do is it can make aggregate solutions based on its computed solution vectors. And so using the solution object has a method called make aggregate solution, where you can provide an identifier for this. And again, recommend doing something SBML compliant that doesn't start with a number, just because it allows for the tab completion and easier access later down the road. And we provide an equation. In this case, we want to look at this occupancy pool, which is the occupancy of the charge the high energy phosphate bonds in the system. And so we know that there's 2 on ATP, 1 on ADP, or equivalently 1 and 1/2. And we can look at that variable or that equation in the solution. We have to provide which variables we're using. We don't have to provide it, but it will speed up the process if we provide this argument, because it'll help MassPy detect which variables to aggregate together. We can then take this new solution, it's in a dictionary format, and just add it into our mass solution object. And as you can see, when we do that, we now have a new key for the occupancy pool. So this allows us to make pools quickly and then make pools of pools. So for an example here, to make the Atkinson energy, the adenylate energy charge, then we would essentially first define the occupancy pool. Then we would go ahead and define the capacity pool, which is just the total amount of ATP, ADP, and AMP. And this is also one of our time invariant pools, as reflected from above when we were making the model. And this update equals true is just a way of saying update the solution object with the new pool. So we don't have to add this line of code every time. We go ahead and we can update, and we now have an occupancy and a capacity pool in our solution object. And so now we can make an equation simply just taking the occupancy, dividing it by the capacity, and that will provide us with the energy charge solution. And so if we look inside here, you can see we have the energy charge key in here. And because this is was made in such a way that no spaces, no dashes, it becomes usable with the tab complete option. And so we can just do the solution.energy charge and get the values of the energy charge aggregate variable. So it's a very handy method for forming aggregate variables quickly based on solutions that have been computed already. And so next is the steady state determination, and how do we get to a steady state? And we have a couple of methods for doing this. We're just going to load up a fresh simulation here and call it sim for simulation. And this time we're going to use the find steady state method. And this has 2 key arguments for it. The first is which model do you want to find the steady state for? And the second is the strategy. And in this case, we're going to use the strategy as simulate. There are some other strategies for root finding methods that are provided by libRoadrunner. However, those, depending on the size of the system or the amounts of conserved moieties in the system, it may have a little trouble. So when in doubt, it's always safe to simulate the solution to long points in time, and then look at and see was a steady state found that way. And so if we go ahead and we use the find steady state method, we get these solutions in the concentration solution object. And note that instead of an array of points this time, because find the steady state is just a single set of points, that's all that we have returned to us in here. So once we have those solutions, we may decide that we want to update our model to steady state. And recall that we had originally set this model to not be in a steady state. This is wise because now we can take this found steady state, we can set the model to update values equals true. And then now our model's new initial conditions are no longer the ones that were for the unsteady state, but now they're for this steady state with rounding error. And lastly, something useful we can do is if we want to find a steady state after a particular perturbation, we can pass that perturbation or that disturbance in. And the way we do that is we take the variable we want to disturb, in this case, the forward rate constant of the use reaction, and we could set this directly numerically. However, it's used in some cases, it's more desirable to perturb it by a particular percentage. In this case, we want to increase the forward rate constant for the use reaction by 50%. So we just take this formula, use times 1.5, and it'll go ahead and under the hood, it will determine, okay, this is take what is the numerical value for K use, and then it will multiply it, create the new solution. When you simulate it, you get a new set of values. And as you can see, these values are essentially they're different than the previous steady state. They're for increase in the solution, in the use of ATP. And so this leads into the final section of this exercise, which is just running perturbations on a model. And there are various types that we can perform. Just to go through a couple of them. Essentially, these perturbations are just another way of saying some sort of disturbance to the model at time equals 0. And the way we do that, well, first, we want to make sure that our model is at a steady state, because more often than not, we're interested in taking our model that is in a steady state and disturbing it to see how it deviates from a steady state, and whether it returns to that steady state or to a new steady state. And so one type of perturbation we can run is just perturbing the initial concentration of metabolite. In this case, we can perturb the concentration of ATP, increase it, or just change it strictly to 2.5. So it goes, the original value of ATP, if we were curious, ATP underscore C, initial condition was 16. Oh, well, shouldn't be 16. Should be 1.6, but probably set that somewhere else. But anyways, take the perturbation, we increase it, and we can see perturbation being performed on the model. And viewing the time profile, again, using our initial argument, using our quick view method is useful. We can see that a steady state is returned to. We can also, again, perform this rate constant perturbation where we increase the rate constant by 1.5, view the results as it goes to a new steady state. And essentially, that is how you perform dynamics, how you build models for simulation, how you then simulate them, and a brief introduction on how you can visualize some of these results. And again, something important to note is when you're running these notebooks, specifically them in notebooks, because it's not necessarily a top to bottom way, run, you can jump around and run cells like I did through it. It's always good to restart, run all once you're said and done, just to make sure everything will simulate and go through. And it's usually pretty quick. Into that, you can see we have our results. And so with that will conclude the first exercise. Are there any questions to it?
Zach, I have a question on those last plots. When you're doing those perturbations and you can see that they reach steady state kind of just in time, is that conveniently set based on the T final, or is that something that's automatically detected where the where steady state was reached?
No. So in this case, it was set based on the T final. And to be honest, it's because I'm making this example. I knew that steady state would be reached at this point. But if you don't have it at T final and you just set it to 100, you will see that you still have some curved behavior at the end. And that's actually what the whole idea behind this find steady state method is up here. Is this method will go all the way through when it simulates to find where this value became, essentially where these lines became flat. And so the actual simulate method will not necessarily stop at steady state, because you may not always have a simulation that ends up in a steady state. In some cases, if you're trying to diagnose an instability in the model, you would want to simulate the model to a long period of time and see which metabolites are exploding up or exploding down to help you determine why is this unstable. Maybe I misset a parameter, or maybe I have something off with one of my concentrations. And so so that's why the simulate method is strictly just simulating the model. But for steady state methods, we have the find steady state option. That will go ahead, and one of its key arguments is you can set the final time point here. And if it doesn't work, it'll tell you there's no steady state.
Okay. And under the hood, what is that doing again?
Under the hood, what it's doing is essentially it's taking these final values in the solution. So let's take this one, for example. And what it's doing is it's saying, okay, what are the values here? So these are the final time points, and then these are the second to final time points, and it looks at the absolute difference between these. No, I think I have an extra prime missing. Is there absolute difference? And then what it does is it takes this absolute value and it compares it to a tolerance threshold. And this is a configurate. This is an option that you can configure. And if it's lower, if it meets a certain criteria, like these two, they'd be considered to be in steady state. If it doesn't, then it's not considered to be in steady state. And this criteria is based on if you set a mass configuration at the very top of the notebook, or not, a little bit more advanced, but essentially, if you say, I want the steady state threshold to be 1e to the minus 10, then it'll use, well, I guess it'll use this value in determining what it is. And so I believe the default is looking for is the absolute difference within 1e to the minus 6. It tends to be pretty accurate for the absolute difference here. But there are, and this is just for simulating them. Using one of the root finding methods, it will look at the the least squares. And unfortunately, the root finding method has a little trouble on this system just because of the buffer molecules being a conserved moiety. But that's handled by libRoadrunner. So that's why we go straight for simulation on this case. But either way, as long as this value is below a certain threshold, it's considered to be 0. As you can see, just looking at these values numerically, they're pretty small.
Got it, thanks.
Mm-hmm.
Any other questions for Zach?
Yeah, just a general question about the MassPy. It looks like a great tool. I think that can be pretty useful. Yeah, is there any functionality or method within MassPy to estimate parameters based on data of fluxes, metabolite concentrations, etc.?
You mean, well, so there are methods for calculating the perks currently, and there are also some sampling methods for concentrations and for and it can utilize the Cobra flux methods as well. Some of the parameter fitting methods are done through the MassF package, which I believe Dan will get to later in the lecture. And there are others that are in progress for, there's one in particular that I'm currently working to develop and add into MassPy. It's called K-cone analysis, which is essentially a type of convex sampling on the perks themselves to try and provide a bit more accuracy there. And get more perks that are more likely to be used throughout an ensemble, and you can see where those parameters converge to, and if there's a lot of variance or very little variance. However, as this is still MassPy is still in its initial just published and still being developed, features are. There are plenty of features to add, and so if there are any features that you could see yourself using or would be interested in, please don't hesitate to come contact me and talk to me about it. Love to look at putting them in.
Cool. Yeah, that sounds great. Actually, I have one in mind, just real quick, because you mentioned also combination, and well, MassPy is based on CobraPy somehow. Do you think or plan to establish like a dynamic flux balance analysis where you use MassPy to model some extracellular metabolites and then use CobraPy to update some exchange reactions based on these?
So at some point I would like to do that. However, actually the newer versions of CobraPy have a dynamic flux balance analysis method included in them. And so right now it's because CobraPi has that, it's a little bit less of a priority, but it's still important to eventually have the two of them kind of synchronize. But that's the issue, is it's not like it's rewriting a method from scratch so much as making sure it's going to stay compatible with the currently existing Cobra method. But yeah, I'd be happy to point that out. I believe in the CobraPi documentation.
Yeah, actually, I didn't know that they have a dynamic flex balances method, but yeah. I believe like since you went through this tutorial now, I believe that with MassPy you would have more flexibility on defining the dynamics around your cell, let's say. But yeah, I would also have to have a look at CobraPi actually at this method.
Yeah, and ideally, again, there's various ways you can do these kinds of methods. Dynamic flux balance analysis could be used to get a flux state that's then used to create a steady state model using MassPy. And so the combination of the CobraPi and the MassPy package together becomes pretty powerful, and there become a lot of various ways of doing similar tasks. So it just, yeah, at that point, it's just a matter of ensuring that they stay compatible. That's the at least from the developer side, that's the big problem. From the user side, if you can get your values one way or another, that's what matters.
Yeah. Cool. I stay tuned about MassPy.
Awesome. With that, if there are no other questions, I will turn off the screen.
Okay. Thanks again to Zach for that excellent introduction into MassPy. So the example there was on a simple chemical reaction, ATP reacting. And I think a lot of the first part, we were talking about these kind of generic small molecule reactions, X1 to X2. But when we're modeling metabolic networks, really we're talking about enzyme reactions, right? And we talked a little bit about the Michaelis-Menten reaction, but we haven't really gone in fully into how do we model enzyme mechanisms, how do we parameterize them, and that's really the topic for this second part. So I already showed this type of reaction coordinate diagram, but just to reiterate these concepts. So we have on the y-axis the energy of the reaction complex versus the reaction progress, going from reactants to products. And we mentioned how in thermodynamics, we're really just concerned about this gap between the initial state of everything versus the final state of everything. But with kinetics, what we're talking about is getting over this activation energy. And so the idea is that whatever the middle state is of the reaction, where groups are being pulled off and shuffled around, is a very high energy state. When high energy meaning it's very unlikely to observe this state. It's a very rare state, meaning it's a very slow process. What enzymes are generally thought to do is to help stabilize that really high energy state, to lower this activation energy, which means that you need less energy, less thermal energy to pass over it. And so the rate will happen, there will be more collisions that have enough energy, and so the reaction proceeds at a higher rate. And how they stabilize this transition state depends on this configuration, how the enzyme orients the metabolites and shifts groups around, local pKa of different residues. So there's a lot of biochemistry involved with how exactly that happens, but kinetically, that's the concept. Is that what the key event is this metabolite binding, and kinetically, that's then very important to capturing the mechanism. And so, typically, you have an enzyme with an active state, and it binds a substrate, and then converts it to products. Maybe water could come in. It's like a hydrolysis reaction. This case is highlighting a hydrolase reaction, but there's different reactions with different kind of inherent classes of or very, very canonical kinds of reactions. So, oxidoreductases might involve some kind of NADH or redox cofactor transferases. And so the type of reaction is going to motivate a very diverse array of enzyme mechanisms, right? You're going to have some that involve cofactors, some that are just a single metabolite, like an isomerase. And so we have to be able to model very diverse mechanisms in order to capture the kinetics of these reactions. This is just a snapshot of what some of these mechanisms look like. So at the very simplest level, we have single substrate kinetics. That's that Michaelis-Menten. Here we have in the ultra simple area, Michaelis-Menten single substrate S to P it was an irreversible mechanism. But in reality, we can't deal with irreversible mechanisms very often because products are present. And as Zach already mentioned, every reaction is reversible at some point of product levels. You're going to have enough product that the reaction will go in the opposite direction. So really, we like to model things in a reversible way. And the majority of reactions involve two substrates somewhere. And so we have to deal with the variety of mechanisms that can pop up. And really what we're dealing with, very broadly speaking, is either sequential mechanisms where you have one substrate binds, then another binds, and then there's some kind of reaction that occurs, and then the products pop off. Or another broad class is a ping pong mechanism where one will bind and react and modify the enzyme itself, and then a first product will come off. So you have like a donation of a group to the enzyme, and then the second substrate interacts with this modified enzyme and then kicks off, and then once again kicks off the actual product of the reaction. So it's, even though it's they're both A and B to P and Q, the actual mechanism is different, and we'll want to capture that kinetically where we can. And even though this seems kind of like a conceptual mechanism, this is actually backed up by biochemistry. If you look at like the transaldolase reaction, which is one of the canonical ping pong reactions, you can actually see which residue in the active site receives a donated group from the first metabolite. So this is all, generally speaking, backed up by the actual biochemistry going on within the enzyme active site, even though we can just represent it kind of schematically with these very kind of black box kind of representations of, we don't like actually have to say this is residue X. And so there is some kind of coarse graining here where we're not modeling every single protonation event within an active site, but we are modeling what we call like a rate limiting step, which are the key events occurring that show the interaction of the enzyme with the environment, right? Because we don't necessarily care about all the internal enzyme events going on within catalysis. What we really care about is these key interactions, so that we can model this in a mass action way. And yeah, so we have quite an array of mechanisms at our disposal to capture that diversity of enzyme catalysis. So just to cover this on the simplest spectrum again, this is our irreversible single substrate Michaelis-Menten. We have our substrate, our enzyme, forming this complex and forming a product in the enzyme. And then typically we think of this binding step as fast and then the catalysis as slow. This is largely borne out. There's some recent surveys of enzyme kinetics that find this to generally be the case, that the complexation is not the rate limiting step. So this is relatively fast compared to catalysis. But we can make a stoichiometric matrix for a system. It tells us we have 2 dynamic degrees of freedom, and therefore we need to define conservation in order to solve this to steady state. And we can define individual rates for these different steps. So our V1 here is K1 times S times E, which is just the mass action on this side. V minus 1 is K minus 1 times X, so mass action on this side going that way. And V2 is K2 times X, so mass action going this way. And there's no V minus 2 because it's considered irreversible. Why do we consider this irreversible? Well, that's a historical fact motivated by the fact that this equation was developed to analyze in vitro enzyme experiment data. And in these experiments, they would put in the enzyme with the substrate, and there'd be no product present, and they would study the initial rate. So initially, there's no product there. So you can neglect any kind of reverse flux because there's no product there to catalyze any reverse flux. So for initial rate enzyme experiments, this rate law is fairly applicable. In vivo, this rate law is often very inapplicable because you're going to have plenty of product there, and this reverse flux is not going to be negligible. So this is really intended to be an in vitro rate law. Just for a little historical context. So we have our dynamic mass balances on each of our metabolites or each of our species. So we have S, dEdt, dxdt, dPdt, and then production and consumption of each. We can also define two different mass balances, one on our enzyme. So we have an enzyme total, that is the free enzyme plus this complex enzyme form. So this X is the ES complex. So that's also where a lot of the E is. So that they sum together to make E total, which we also call E naught. And then our total substrate is in S plus X plus P. That's the S total. Okay, so how do we get from these mass balances to the Michaelis-Menten? Well, the key assumption is that we do a steady state on our intermediate complex. So we say that our intermediate complex X is not changing over time. So we do a steady state mass balance on that complex alone. So now we have our dynamic mass balance again for this complex, which is the same as there. But now with our enzyme total subbed in. And so once we sub that in, and we get we solve it at steady state. So we set dxdt equals to 0, and we solve for X. So now X equals, you can just solve it out here, E naught over S over this. And then we plug this expression for our steady state X into our product formation mass action equation. So this dPdt, which is here. So we plug in X here, and you can see we end up with this expression, which once you call this K2 your Kcat, and this expression your Km, is our Michaelis-Menten expression. So in other words, it's a very simple derivation resulting from mass balances plus a conservation expression on our total enzyme plus a steady state assumption on our complex formation. So that's why it's this quasi steady state assumption. And this Michaelis-Menten equation is very important because it exhibits some of the key properties of enzyme kinetics that differ from chemical mass action reaction kinetics. The first is saturation. So if you just have a mass action reaction, what we said was that it's proportional, the rate is proportional to the concentrations. So if you keep adding concentrations, you'll keep getting more reaction rate with no inherent limit, right? However, the Michaelis-Menten equation saturates. So as you add more S, you hit a Vmax, you hit a hard upper limit on the reaction rate. Why is that the case? Well, that's because you have a fixed amount of enzyme that's catalyzed in this reaction. So the other inherent assumption is that you have an E naught that is fixed. And so if you consider E naught to be variable, then yes, it'll be unbounded again. So if you keep adding enzyme as well as substrate, then it'll be unbounded. But typically in a experimental condition, we'll be having a fixed amount of enzyme and changing our substrate, and you'll get these saturation kinetics. So that's the first parameter, is what is the Vmax, which gives an idea of how how fast this enzyme can catalyze a reaction. And then the other parameter is the Km, which has an interpretation as of the substrate at half max velocity. So it's almost like a binding constant where it tells you how much substrate do you need for this enzyme to be fairly active. In this case, half active. So this is Vmax over 2, and the K at this point the S is equal to Km. So those are our key parameters, and this is our classical interpretation of Michaelis-Menten. So you can absolutely build a kinetic model full of Michaelis-Menten equations. People often do that. The potential problem there is that you've made many different quasi-steady state assumptions in the process of doing this. What makes an enzyme okay for quasi-steady state internally is not necessarily going to be consistent across enzymes. So a lot of your assumptions might end up not being correct or might not hold up once you start to link these quasi-steady state networks together. And then also, as I mentioned, this is really an irreversible system. So at least you'd want to use the reversible form of this, and which is not too terribly different than what this looks like. So there is a reversible Michaelis-Menten that you could potentially use. So that's certainly one way. So you can think of it as there's two ways at face value to model enzyme kinetics. One is to use reversible Michaelis-Mentens, which have a bunch of quasi-steady state assumptions built in as a downside, but the upside is you get these overall rate laws that are nice and interpretable and very simple. And the other way is to leave your system as mass action and just model these individual mass action reactions. And the challenge there is that you have more parameters to parameterize. So even though this just has Km and Kcat, this now has all these individual rate constants that you have to specify. So two contrasting approaches, both having their advantages and disadvantages. In our lab, we mostly go with this approach because we have a specific package for parameterizing these individual rate constants called MassUp that I'll go over later, which helps us get around the downside of this. So it's kind of all upside. So we have these nice simple mass action equations, and we don't have to deal with any incompatible quasi-steady state assumptions. So enzymes get even more complicated than this. Maybe I'm not going into all of the different varieties of complications. For example, I'm not talking about different inhibition schemes of enzymes, which there's a variety, but a really critical one to talk about is cooperativity. So in the Michaelis-Menten typically has this kind of hyperbolic curve where you have this saturation behavior. With cooperativity, you can get a sigmoidal curve, which looks instead like an S. You might be familiar with field kinetics. And cooperativity really captures the fact that as one, different effectors can, as they bind to the enzyme, change the likelihood of other effectors binding the enzyme. So what can happen, for example, is as an activator binds to a complex, it makes it easier for more activators to bind. And that results in switch-like behavior, which is really critical to the regulation of certain enzymes like PFK, PYK. A lot of these key glycolytic enzymes are cooperative. And so we need a mechanism to account for that. And there's several out there. And one of the more prevalent models that, again, has been shown to be sort of correct by biochemical analysis for many enzymes, at least, is this Monod-Wyman-Chengo MWC model called the symmetry model. So how does this work? How does this differ from Michaelis-Menten? So once again, we have our simple S plus E goes to P plus E reaction, but now we're modeling an inhibitor that cooperatively inhibits this enzyme. And this model is based on a mechanism that requires the enzyme to be multimeric. So a multimeric enzyme has multiple subunits. Each subunit can catalyze the reaction, and each subunit also has an inhibition site. And the key behind this mechanism is that the each subunit can also be in two forms, an active form, which we call the relaxed form, or an inactive form, which we call the tense form. And it's a steric change in the shape of the conformation of the enzyme, such that it goes from, it's always bouncing back and forth at some rate between the active form that can catalyze reaction and inactive form that cannot. Now, the basis for this model is saying an inhibitor can only bind to the inactive form. And so when an inhibitor binds, it goes from an inactive unbound to an inactive bound for one of the subunits. And it can do that at each of the subunits. And so you can have 0, 1, 2, 3, or 4 inhibitors bound. Now, how does this create cooperativity? Well, as one inhibitor binds, it actually locks the other 3 out of being in the active form. So as one binds, it makes it easier for the other, for more inhibitors to bind, because the other 3 sites are now available for more binding, and they're unavailable for catalysis, right? So in other words, you have cooperativity, whereas one binds, it makes it easier for the next to bind, so on and so forth. So it creates a switch-like behavior in that as soon as any inhibitor binds, you get a very rapid response where all the inhibitor will bind and the enzyme will be completely locked up in this inhibited form. So that's the basis for this model. For activators, it works the same way, except it locks it in the active form. So it locks it in the R form instead. And there's even substrate cooperativity, where substrates can bind to the active site and lock it in the active form. So cooperativity can show up in a lot of different ways. And this isn't the only way that cooperativity can show up in enzymes. It's just one of the models that has been more successful and works for a lot of different enzymes. And we can go through the same kind of procedure we went through with the Michaelis-Menten, deriving the mass balances in our conservation equations. And we can get a rate law that accounts for symmetry now that looks like Michaelis-Menten, but has a couple additional parameters. One is this L, which is the inherent favorability. It's this the KEQ for this the transition between R and T with nothing there. So like an inherent preference for one form versus the other. It has a KI, which is how the binding constant for the inhibitor. And then it has a coefficient that creates this sigmoidal shape, which has to do with the shape of the how many multimers you have. So in this case, we have 4. So we can account for these, and again, once again, just like with the Michaelis-Menten, we can choose to either use this overall rate law or just use these individual mass action reactions. Once again, you just have this explosion of parameters that you have to be able to deal with. While here you have a bunch of built-in assumptions that you have to be okay with. So that's how we can model these more complex enzymes. And then at the ultimate level, we can talk about building enzyme modules which capture all the known mechanisms. So it could be an MWC model plus pH or plus other activation or inhibition going on. So as you layer these mechanisms on, you can describe very complex reactions just in terms of mass action. And we call this group of individual mass action reactions, we call that an enzyme module. And so here we might not have a textbook rate law like this that has been derived, but we can still model it as long as we can model the individual mass action reactions. And this often will mirror the biochemistry going on, as I'll go through for the PFK example. Just some more on how complex these mechanisms can get, but we can still account for. So conditions also affect enzyme activity the same way that they affected the equilibrium constants. We talked about how certain reactions are pH and temperature sensitive as far as their equilibrium constant. They also affect the kinetics just as well. For example, enzymes are well known to have an optimal pH. So as you decrease pH, they get less active. And as they increase pH, they get less active. And it's not always clear exactly why that's happening. but we can model that by approximating that there's a critical deprotonation or protonation step that's happening that's decreasing the activity. So as the pH decreases, you're adding more and more protons to your system, and at some point it might protonate some residue that destabilizes a binding site or makes it hard to catalyze the reaction for some reason. And that is leading in this model to the decrease in activity, and vice versa on the high pH side. And so we can model that again in a mass action fashion by saying, okay, there's the correct form of the enzyme, which has an approximately correct pH state. And then you have either protonation or deprotonation events that hurt the activity. And so you just add these binding equilibria along with dissociation constants, they're the pKa's of these residues, whatever these key residues are. And you can fit this to this kind of curve and approximate what those key pKa's are. And so you never really identified what the residue was, but you were able to capture that behavior. In some cases, you can actually identify that residue through substitution mutagenesis and different actual experiments. So again, this has been borne out by biochemistry, and there's a really nice chapter on this in that textbook I mentioned, the Methods in Enzymology textbook, Contemporary Enzyme Kinetics, on this. But this is just to show you that these complex processes can still be modeled to some degree of resolution. And same with temperature. We all know that reaction rates very generally increase with temperature because molecules have more thermal energy, so they can get over that activation energy more often. And this has been approximated with transition state theory, where you break your, you in general can approximate your rate constant as a function of temperature by modeling the activation energy. And even further, this can be broken down into an activation entropy and enthalpy. So like, you know, there are models that capture this that you can apply. We've done some of this, but generally we don't have the degree of data necessary to justify this kind of expansion. But this theory does exist, just to give you some idea of how far this goes in terms of modeling these things precisely. So let's step through a case of how this works for a real enzyme. PFK is a key enzyme in glycolysis that forms fructose bisphosphate from fructose 6-phosphate. So this priming of this metabolite before it's broken down into triose phosphates to capture energy. This is a key, thought to be at least a key step in glycolysis, a rate limiting step in glycolysis. And so it has a lot of regulation. It's a tetramer, which, as we just mentioned, enables it to be cooperatively regulated, which indeed it is. In humans, it has 3 different isoforms, which would further suggest it has diverse regulatory roles. And it is cooperative. So it has these 2 natural conformations that have been structurally characterized. So people have done X-ray crystallography on PFK to actually visualize these 2 different complexes, which bears that the MWC model is actually, you know, backed up by biochemistry. It's not just a thought model or something that works out mathematically. And so there's this, because it's cooperative, you would think that it's regulated. Indeed it is. In fact, there's actually, like, you could think of it as a tug of war between inhibitors and activators that all act on this complex. So in humans, ATP citrate PEP, and AMP, and all these compounds are known to regulate it. And so they're kind of pulling it between these 2 forms. And then we also, of course, just have its catalytic activity and probably some substrate cooperativity as well with the substrate binding. So just to give you some idea of how it's bound, you have the tetrameric complex. You have an allosteric site, which is again, or allosteric sites, which are stabilizing or destabilizing the active form of the enzyme. And you have catalytic sites as well, and multiple because it's a tetramer, and the key residues have been identified. So this very actually closely models the MWC model that came up with, as a conceptual model well before this. So how do we actually model this? Well, we can do an enzyme. So because the mechanisms of activating and inhibiting this enzyme are so diverse, it's really one of those cases where we want to build one of these flexible mass action enzyme modules, because then we can just layer mechanisms on top of it and model it with mass action, because it's very straightforward. So we have here the catalytic tracks, and there's multiple active sites. So we have multiple different tracks that can all be active. We have the RT transition. So only the R state is actually reacting. So that's why the catalytic states come off of the R state. We have the R to T transition that models this activated to inhibited structural transition. And then we have activators binding the R state to stabilize this form. And then anything with activator bound can still catalyze the reaction. Anything with inhibitor bound is locked into the T state and cannot catalyze the reaction. So this is an example of one of these enzyme modules and how mass action can really enable us to model very complicated mechanisms without really a lot of headache. The only headache is in finding rate constants for all those, and I'll go into how that's done. And you can see it becomes an absolutely massive reaction network just to model a single reaction. And that's another limitation, is that these things get very large and complex. And then there's different pathways that you can derive from this. This is more steady state kind of analysis, but there's different reaction paths that can be analyzed in the S matrix, the stoichiometric matrix. This is the null space of it that model the different catalytic paths through the enzyme. You can also define key pools. So all of the forms of the react form. Again, the reason you would define pools is because it's very nice to plot. So if you have hundreds of variables just to represent one enzyme, you really want a snapshot of what that enzyme activity is. And so by defining these pools, you can plot these aggregate variables instead. And hopefully that provides you that snapshot of, okay, there's a big transition between R and T as you add this substrate or something like that, right? Like you really have to find some way to simplify these models to plot them. And these aggregate pools are a great way of doing that. So you can plot the R and T forms, you can plot just the catalytic state, and you can plot key ratios like fraction of the total enzyme that's in a particular form. Once you plug in some kinetic parameters into this, you can find, for example, that most of it's about 90% is in the R form, but it's only about 12% of its catalytic activity, which suggests that maybe there's too much inhibitor present or something along those lines. Okay, so now get into how do we actually parameterize these models, and that gets into another package that I don't have time to go into fully today, but it is also publicly available, and you're free to check it out if you know Mathematica, because it's a Mathematica package. So we've talked a lot about different kinds of parameters, and we've just kind of assumed that we knew them, that we could find them somewhere, that we can approximate them. That's often the major challenge of kinetic modeling is constraint-based modeling. You need almost no parameters. With kinetic models, all of a sudden you need hundreds or thousands of parameters, and like, where do you get them and how are they affecting your model? These are major issues. So there's two kinds of parameters setting this up. There's the microscopic parameters, which is what we've been talking about almost all this time, because we've been talking about mass action. Those are these microscopic forward reverse race constants for individual reaction steps of enzyme mechanisms, right? So if we want to do mass action modeling, this is what we need. But when we get kinetic data, for example, with initial rate data, or we're analyzing those experiments with Michaelis-Menten rate laws, we're not getting microscopic parameters. We're getting what we call macroscopic parameters, things like KMs and KCATs and Hill coefficients and things like that. KIs is another big class. And so there's a big mismatch between the data that we can get for the kinetics of enzymes and the data that we want if we want to do mass action modeling, which is the most flexible way to model. And it's the method of choice, at least for the work we've been doing in the lab. So how do we get to our rate constants? Well, as you already saw for the Michaelis-Menten equation, there is a relationship between the micro and the macro. These are related, they're not independent. And so there are expressions that we can actually derive that explicitly relate these. And then, so the goal then becomes, okay, let's define those expressions and then find rate constants that satisfy the observed data, the observed kms and kcats. So that becomes a parameter fitting or an optimization problem. And that's the basis for MassF. So our strategy in this lab for parameterizing these enzyme modules is to take this kind of kinetic data that we have and map it onto the microscopic rate constants so that we can do mass action modeling. So just to reiterate this parameterization challenge, these mass action models and the enzyme modules that we've been talking about are really flexible. We can model basically anything, including very complex condition-specific pH sensitivity, inhibition schemes, all that. The people, enzymologists have come up with ways to model all these processes. And they're also mathematically simple. They're just, they're easy for solvers to deal with. They're generally stable because they have kind of a spring-like capability. As you get more of something, it decreases the rate. So they're very stable, relatively speaking. But they have a lot of parameters, and there are these microscopic parameters that almost no one measures directly. There are a few ways, but generally speaking, we're not measuring microscopic rate constants directly. And so we're almost never going to have full data, meaning we're going to have way more parameters than we have individual data points. So you might be mapping 1 km and kcat to 10 parameters. So like, is that even a viable way to do modeling, right? That's a challenge. And so there's going to be uncertainty in our parameters, and we have to be able to account for that uncertainty while still matching the data that we do know. So that's the challenge. And so the MassF package that we developed in the lab is a workflow to do that, essentially, to take our km and kcat data, anything we can find, the macroscopic data, and extract from it all the microscopic rate constants in a way that they match this data and also account for the remaining uncertainty, the underdetermined nature of our system. So the MassF workflow is located in GitHub. It's part of the OpenCobra project, but it is all in Mathematica. So so this is a Mathematica workflow. And why is it a Mathematica? Because Mathematica has really powerful symbolics. This relating the macro and the micro requires a symbolic solution. And that is best done still in Mathematica. Once SymPy and Python symbolics catch up, I think we'll probably make a transition to a fully Python workflow. But for now, we do this in Mathematica. And this workflow, the MassF workflow, which is mass action enzyme fitting workflow, is a workflow to first build these enzyme modules for individual enzymes. So we build this module and then finds the microscopic rate constants K. So that's all it does. It's a K finding workflow. It's built on another Mathematica toolbox that our lab developed, Nico Sonnenschein, called the Mass Toolbox, which was the predecessor MassPy, which is the kind of the Python version, or that's how it was initially conceived, was the Python version of this Mass Toolbox. So this is another kinetic modeling package, but in Mathematica. And there's essentially 5 steps in this process to extract K from your macroscopic constants. First, we import the data. We go in the literature and find all of the macroscopic kinetic data for any enzyme we can find. Then we define our enzyme module. So this is specifically going to capture any known mechanisms. So if it's allosteric, we model that. If it has inhibitors, we model that. We generate a reaction scheme for it. Then the key step is we generate functions that relate our microscopic parameters to our macroscopic. So we create functions that say, okay, this Km is a function of K. Then we plug those functions in a least squares into an optimization package to fit these rate constants and evaluate whether it works, whether we are able to find rate constants that fit all the data. Okay, so let's step through these steps. So first, we just curate all the data. So we import it and then curate it so that for every enzyme, we have a number of kinetic parameters that all have conditions, they have references to the original study. We just keep this in a big table for now, and we import that into Mathematica. So this is a Mathematica snapshot. And as I mentioned, our macroscopic parameters that we're typically dealing with are Kms, Kcats, Ki's, Keqs. They come from sources like Brenda. A lot of it's from the literature. Keqs come from either the NAST, the enzyme catalyzed reaction database, or from group contribution, as I mentioned before. And we do some temperature and ionic strength correction. Then we define our mechanism. So we might have a catalytic mechanism for GAPD that looks something like this. Just one substrate binds, the other substrate binds, it reacts, and then releases the products. And we create the same mechanism in Mathematica and build an enzyme mechanism out of this. And we can account for allosteric inhibition schemes, and we create a mass toolbox model object now because we're in Mathematica. Now the key part here is we generate comparison equations from this model. So we find some kind of function that is a function of our rate constants, our microscopic rate constants, that approximates our data. So this could be a Km, a Kcat, or something. For example, we know that the Kcat of our data is the Vmax, right? That's something that Vmax over E. So as long as we have an expression for Vmax, which we can derive from our rate law, that's a function of Kcat. We're now relating our microscopic Kcat to our microscopic K to our macroscopic Kcat. So this equation creates that micro to macro relationship, and all we need to do is ask a solver, tell me a K that solves this, right? It's just a K fitting challenge then, because this, we already have this value, we have this value, we have this value. We just need to ask what is a K that fits this. And oftentimes this involves simulating curves, and you can think of it as a linear regression where, a nonlinear regression, sorry, where you're asking for Ks that cause all these curves to get fit by your model. So how do we solve this? Symbolically, we solve these systems just with Mathematica's solve function. Historically, you'd have to use a King-Altman steady state finding algorithm, but Mathematica's solve computer algebra system is so powerful that it can kind of do all this for us under the hood. So we ask a lot of Mathematica, and that's kind of why we're stuck there. So how do we actually fit it then? So as I mentioned, the challenge here, what we want to do is to find Ks that minimize our sum of squared deviations. In other words, find Ks that cause our functions to fit the data. And that turns out to be this kind of least squares formulation, but because these functions are nonlinear, it's nonlinear least squares. It's a very challenging problem because these functions are extremely nonlinear. For the very simple Kcat or Km definition is a very simple expression. For real enzyme modules, these are massive, massive expressions. And when they're highly nonlinear, you have to deal with the fact that you're going to have local optima. You can't guarantee that you can solve this efficiently. So numerically, it's very difficult. And also, your Ks are going to be spending many more orders of magnitude, which means you're going to have numerical performance issues. So to deal with this, to solve this in an efficient way, we use a two-stage approach. The first is we use a non-derivative based, very robust method called particle swarm optimization in order to find initial points that are kind of okay. So like kind of the start of some of these valleys. So particle swarm does that. And then we plug it into a very nicely performing derivative-based method called Liebenberg-Marquardt that's very robust to some of these issues to kind of finish off each point and optimize it to very close to its local minimum. The other feature is that particle swarm is random. It's a random method, so it'll give you a diverse array of starting points. So you'll get an idea of the uncertainty in your value because of the randomness here. So it actually addresses that other issue I mentioned, that because we have an underdetermined system, there's alternative optimal solutions. And so this PSO is a way to get those alternative optima. So this Liebenberg-Marquardt algorithm is this damped least squares. This is derivative-based optimization. So it's very good at finding local optima using gradient descent. And it also has this damping parameter that helps with poor scaling systems like ours. So it moves really fast in flat regions. But we tried using just this method and it still didn't solve because the starting points that we would randomly choose were still so bad that it didn't find a good fit. So we had to find some way to provide it with better initial points. And that's why we started using this particle swarm. And particle swarm is a randomized method. It just kind of looks around this parameter space and uses information from its neighbors to find areas that are somewhat decent. So it's trying to find the star here, and they would find all these points would use information from points that are near the star to say, hey, there's a star over here, and they would all end up over there. And so this is a very robust method to find initial points, but it's very bad at doing the final optimization. And so we pair these two together to kind of address all our problems here. And just briefly, so once we actually fit it, we get our rate constants out of this, and we can assess the goodness of fit. We can cluster alternative solutions to see if we're finding kind of characteristic points, and we can assess parameters uncertainty. Okay, so that's how MassF works, and we end up with our rate constants. So in the exercise today, we're not going to be actually running through MassF, but we will be importing modules that were fit with MassF. So that's how we, in the lab currently, we are getting our microscopic rate constants to get enzyme modules to import into MassPy and then do all this other work. That's our typical workflow. So just an example of what this looks like for GAPD and glycolysis. We have our mechanism here. This order is suggested by people who study this enzyme. They say it's an ordered mechanism, and this is the binding order. So we can just capture that here. We have our data. So we have the KM that we got from Equilibrator, which is a KEQ website. We have the Qcqa 268 inverse seconds, very typical. And then three different KMs, as well as one KD measurement, so one dissociation constant measurement. You can see it's not complete information. We don't have a reverse Kcat. We don't have reverse Michaelis-Menten constants. So this really wouldn't be enough on its own to parameterize irreversible Michaelis-Menten rate law, right? So, so already we couldn't build any other kind of model other than with some kind of parameter estimation. But when we created the comparison equations, we can see that we have different simulated curves for each data type. So KEQ, the different KMs, and the different Kcat and the KD. And in orange is the model and the blue is data. You don't see the blue because this was a perfect fit enzyme, so they're perfectly overlapping. What you'll sometimes see is that there'll be a line of blue below. That means it wasn't able to perfectly fit this. And that happens when you have inconsistent data. So sometimes we have perfect data, like data that's complete, quote unquote, like forward and reverse Kcats, forward and reverse Kms, and a KEQ, but it's not internally consistent. And so you can't actually find rate constants that match all the data because there was some error in the data, and it can't all be fit simultaneously. And so MassF will find a point that is somewhere in the middle that tries to balance the error on these different data sets. And when we can look at the actual K microscopic Ks on a log scale here for different individual steps. So these are the different individual Ks up here that came out of this fitting procedure. And if we repeat this tens or hundreds of times, we get distributions. And we can see that for some Ks, there's a very wide range, meaning there's a lot of uncertainty. And for others, there's very little uncertainty, meaning they're very tightly constrained by the available data. So what do we see here? We see that on the right side, these are all the reverse reaction steps. You see they're almost all very unconstrained. Why? Well, that's consistent with the fact that we have almost no reverse kinetic data. So because we have no data, we have almost no idea what these rate constants are, and so they end up highly unconstrained. Well, for the forward side of the reaction, they're very well constrained because we have a lot of data in that direction, okay? And so you can see so this is looking at these independently, but they're actually interconnected. So there's nonlinear dependencies between these rate constants that you can visualize in this plot, and you can actually cluster this to get characteristic sets of rate constants that kind of capture the behavior. So if you repeat this optimization with 200 or 1,000 data points, you don't end up with 1,000 different candidate rate constant sets. You can actually simplify it and get characteristic rate constant sets. So that's how this works. You get these rate constants, and then you plug them into Zach's workflow in MassPy, and that's what we're going to go through in the second example. Okay, so very briefly, I see that we're a little out of time here, but I wanted to go through one practical example in literature. Like, what do you get out of doing all this kinetic analysis? Well, we talk a lot about simulation, and it's very useful for understanding the dynamic properties of your system. You can use it for strain design to see how do you increase flux in your pathway. You can perturb your model in different ways and look at what increases flux the most. But I wanted to show a little bit on how you can actually use kinetics for data analysis as well. And so the goal, so I'm covering a key paper that came out about 5 years ago, which is one of the first people I've seen to look at in vivo kinetics from a theoretical perspective. And the question they were looking at is, how is flux controlled by metabolite and enzyme concentration changes in vivo? So we know, or we think that so we can create rate laws that like Michaelis-Menten or like mass action, that relate X and E. But the question is, or a question is, how is V actually controlled in vivo? So does X or E exhibit more control? So how does a cell actually change its flux from one state to another? Like, let's say, like the cell is experiencing a energy starvation, and it knows it has to change its flux state to respond to that, right? How does it actually do that? Does it do it through the through transcriptional changes by changing its enzyme levels, or does it kind of happen naturally through concentration changes? And are they the substrates, or are they the allosteric regulators that are changing it, right? Like, basically, how is flux controlled in vivo? And a more fundamental aspect of this that I find interesting, and because a lot of people are skeptical about kinetic modeling, is can in vitro kinetic rate laws, all these models that we're building based on in vitro data, can they even represent at all how flux is controlled in vivo? Or are we just completely missing how it actually works in the cell because it's such a big mix of different things going on? Maybe everything we're doing in vitro is completely irrelevant, right? And so one of the first studies to do a really nice controlled experiment was this 2016 science paper from Josh Rabinitz's lab at Princeton. They did this in yeast, and they did a study across 25 growth conditions, specifically 5 substrate at 5 different controlled growth rates in a chemostat, okay? So let's take a look at their setup. So they measured 3 parameters. They measured 3 data sets, fluxes, enzyme concentrations, and metabolite levels. So essentially all the variables in a rate law, they measured those directly in vivo. Then here were their conditions. So they did 5 different substrates at 5 different dilution rates or growth rates. So the same substrate at 5 different growth rates at different times. And the question they're interested in is if they take these values and they plug the enzymes and metabolite levels into a Michaelis-Menten rate law to approximate V. does that actually match the measured V, right? So does the left side of this equal the expected right side based on our understanding of kinetics? In other words, do the kinetics work? And then do the kinetics explain how flux is actually regulated in vivo? And then what they did further beyond that is for cases where it doesn't look like it works, can they make it work by accounting for previously unaccounted for regulators and factors that we can actually add into a new Michaelis-Menten rate law? So let's look at a case that works first off, which is this TPI reaction. Very simple glycolytic reaction going from dihydroxyacetone phosphate to glycerolide 3-phosphate. And you can see the metabolite profiles on different substrates and different growth rates. So DHAP at different dilution rates. So this is not dynamic, this is steady state, but it still can be analyzed with kinetic models. And then you have the actual rate, the rate of the TPI reaction. So they took these values, they plugged them in a Michaelis-Menten expression for the reaction. So this is that very simple, this is the reversible Michaelis-Menten now that I was alluding to. And they calculated the flux and they compared it to the actual flux, right? And this is what they saw. So you could see a pretty good match between the expected flux and the real flux. So the real is in blue and the expected, the measured, like the Michaelis-Menten is in red and the measured flux is in blue. And it matches pretty well in all cases, meaning that for this reaction, they have a pretty good idea of how this flux is controlled. It's controlled by the substrate level. They also measure, oh, sorry, this is not the flux activity, this is the enzyme concentration. So they plug in the enzyme concentration as well. So accounting for these three factors is enough to actually model the flux regulation. So in other words, for this enzyme, they are able to explain how its flux is changing in vivo, which is to me remarkable on its own. This would mean that in vitro kinetics are relevant to understanding in vivo kinetics, which a lot of people, I think, were and still are skeptical about. Now let's look at a case where it kind of works, but not perfectly, which is this PRPP reaction. And they did the same thing, measured the substrates and enzyme and plugged it into the reaction. And now you can see, although the agreement still agrees, there are these cases where it doesn't really match up that well on carbon, for example. And what they found is that they were missing a known regulator, which is AMP. And when they account for AMP's concentration change as well, now they got a much better agreement. So now it's agreeing here and it's agreeing there better. It's agreeing there much better on this condition, you can see here. So even just filling in gaps in their own Michaelis-Menten rate law is enough to capture more of the regulation. So this is a case where the regulator actually adds a lot of flux control to the reaction. Now, beyond that, they're like, okay, if we can identify this, we can see that we can observe regulator activity. Can we use this to discover new regulators that weren't known but are involved in flux regulation? So here, what they do is they take the expected values and then they plugged in different candidate regulation events in a Bayesian way, try to predict whether or not a new regulatory event was supported. So in other words, if they try to add random regulators into these rate laws, can they get evidence that it actually helps explain the observed flux change? And this is what that looks like. You have the R squared, how well it worked for different reactions. So you could see, for example, IDH, the existing model doesn't explain well at all how that enzyme is regulated in vivo. So this is kind of like a failed forever case. But they had a lot of cases where initially they didn't have a very good idea of how it was regulated, very low R squared. But when they add a metabolite regulator, it all of a sudden explained a lot more of the flux. So all of these yellow bars are candidate new regulator predictions. And so for each of these cases, you might go and investigate, okay, is this metabolite actually binding to this complex and regulating its activity? And that's what they went and tested. So for pyruvate kinase, they tested fructose bisphosphate kinase, which was a known regulator, and then also citrate, which is a new predicted regulator, and showed that once you account for citrate, the agreement goes way up of this enzyme. So it goes from this 0.5 to accounting for both regulators 0.88. So this is a prediction of the model that adding citrate as a regulator of this enzyme, well, it's the observation that adding it as a regulator makes it fit much better. The hypothesis is that this is actually regulating the enzyme. And they went and tested it and indeed saw that the enzyme is inhibited by citrate. And they showed the activity of the enzyme versus both of its inhibitors now, so fructose bisphosphate and citrate. And you can see that they actually kind of independently regulate it, which is really interesting. In some carbon sources, it's regulated by fructose bisphosphate. You could see that fructose bisphosphate is changing, and that's what's changing the activity from low activity to high activity. But on this nitrogen case, it's actually the citrate that's regulating it. So it regulates it from low to high activity. So it's like a condition-specific regulation thing. So really interesting stuff, and also really encouraging for the ability of kinetic models and kinetic analysis to tell you a lot about your data. And so they kind of summarize this behavior, asking what is actually regulating enzymes in vivo. And the thought might be transcription regulation. It's all going to be enzyme level regulation, but that's not what they found. They found that for the vast majority of enzymes, it's just substrate level regulation. So mass action itself is enough to regulate the flux of the majority of enzymes. It's all mass action dominated by substrate level. But interestingly, once you break it down by thermodynamics, so reversible reactions are almost entirely modified by flux modified by substrate level, a little bit by enzyme. But the irreversible reactions, which are the ones that are insensitive to substrate and product changes, are actually much more regulated by transcriptional and allosteric regulation. So there is a place for these other canonical mechanisms, but it's mainly for these thermodynamically irreversible reactions. So really deep understanding obtained about flux regulation in vivo from this allosteric, from this Michaelis-Menten analysis of these deep in vivo data sets. So that I believe is my last slide. Thank you all for listening. So for the second exercise, we're going to cover a more realistic workflow for building a pathway model with enzyme kinetics. And so Zach Heyman's going to step through the first part, and I'll step through the second part. And this is very typical to the workflow that we're doing. For example, we're building a tryptophan model and using this workflow and importing these enzyme modules from MassPy, or sorry, from MassF into MassPy to get these rate constants, and then using MassPy to simulate and analyze these models. Okay, so I'll stop sharing now and let Zach take over with part one of the part two exercise. So if you want to follow along, this is going to be module 2, part 2 exercise. And the first thing we're going to be going through is this COBRA to mass model workflow. All right. Thanks, everyone.
All right. Should I just dive right into it?
Yep, I can see your screen, so we're all good.
All right, cool. So just quick little background. In order to do this workflow, we needed some data obtained experimentally. And so we got some growth data for glucose media, and that was obtained from these two papers. I'm not going to go through the papers for sake of time, but essentially, if you're interested in where the data came from, the links are at the top. And so what we're going to do here is we're going to use, we're going to take the E. coli model, IML 1515, and we're going to build a subnet work for glycolysis, and then essentially replace every reaction with an enzyme module fitted by MassF. And so we do this and note that we will need some sort of optimization package that can do quadratic programming. So Gurobi is a great package for this. We're just going to disable the output of it so it's not crowding up the notebook. And so we go ahead and we set Gurobi as the solver. And we go ahead and load the Qcqa model for IML 1515. And once that model is loaded, we go ahead and load our flux data as well. And this flux data is in a separate data sheet right now under the sheet name flux data. And it's for the glucose growth medium, but you can do this for an abundance of different types of growth medium for comparison purposes. But for now, we load the data, and these are the fluxes that we have available with data points. And we go ahead, and in this particular case, we set the growth rate to be at a fixed value and the medium uptake for glucose to be at a fixed value. And sometimes this is a little restrictive, sometimes it isn't. In this particular case, it's not an issue. And so we go ahead and we set these bounds, fixing the biomass, fixing the glucose uptake. And then we formulate a QP minimization problem, essentially, to minimize the adjustments on the data with respect to the original, to minimize the adjustments of computed data with respect to the original data. And specifically, we pull out the two irreversible enzyme pairs in this network. Because it's glycolysis, we have PFK and FBP irreversible enzymes that kind of work opposite of each other, as well as the pyruvate kinase and PPS enzymes. And the reason we do that is we tend to get flux data measured as the net flux between these two. However, setting the flux for one of these enzymes as 0 will cause no enzyme to be formed in some cases. And so just to be on the safe side, we can know that these enzymes are present in the model. We'd make a slight adjustment to the flux data, keeping the net flux the same, and just adjusting them by a small percentage. In this case, we assume that the flux through the opposite enzyme is about 10% of the flux. And so we make the adjustment, and we go ahead and we optimize this model using the quadratic programming to minimize the data adjustment. And we get this new set of fluxes, and they're for the entire model. And what we can do is we can compare those fluxes to see how well did this fit. And as this 45-degree line is essentially a perfect fit. Anything that shows up on that line means that there is the data that was measured experimentally ended up being the adjusted value. And as you can see here, most of the data fit really well, with the exception of this one particular dot, PGI, which might indicate that if you look here, the flux was adjusted for sign, which might indicate a data sheet error, might indicate something else, but just an interesting thing to point out and something to look at a little further. And we go ahead and we use these fluxes to create the mass pi model of it. So we just create a mass model of glucose, and we extract the reactions we're interested in. And specifically, we're just interested in the glycolytic pathway this time. And so we extract those and we add them, extract them from the Qcqa model. We can add them to the mass model as mass reactions simply by just taking a Qcqa reaction and popping it into the constructor. And now we have a mass model with 19 reactions, 12 metabolites, and 19 metabolites, 12 reactions, and no initial conditions defined. So next, we have to convert our units because our flux data tends to be in grams. As you can see, millimole per gram of dry weight per hour, and our concentrations are in moles per liter, typically speaking. And so we perform a conversion using some assumptions and some volume data from the second resource at the top of the page to get the fluxes in terms of moles per second. And from there, we take equilibrium constants that were extracted using Equilibrator, using a group contribution method from Equilibrator. And we set those for the reactions. That's what this line is doing here. And lastly, we take the concentration data from the data sheet and we load that. And the concentrations, as you can see, are provided in moles per liter. And so it's a good thing to make sure that we keep our units consistent, as that can lead to a lot of issues. And lastly, when we added the reactions to the mass model, it added all of the metabolites associated with those reactions. However, for the PFK isozyme, or the first isozyme PFK, we have a the activator GDP. And so we have to add that metabolite manually. And again, it's an easy conversion. We just take the Qcqa model metabolite, we pop it into the mass metabolite constructor method. And specifically because we don't have GDP flowing in or out of the system, we just set it as fixed. And what this does is it prevents a differential equation from being generated for it. Or it specifically sets a differential equation to 0. When you integrate that, it's a constant. So it keeps this value constant in the simulation. And from there, we're going to go ahead and we're going to load our growth data into the model, the mass model, just iterating through the data frame. And we also set hydrogen and protons and water. We set those to be fixed as well, because modeling them, we tend to ignore them, modeling them, unless we're trying to explicitly model them for some reason. And we set their initial condition to 1 to kind of make sure they get factored out, at least mathematically. Anything times 1 is still the original value. And lastly, for few metabolites, we don't have data for them. Specifically, we don't have data for phosphate, G3P, and lactate. And so what we do is we set an initial guess just to help the solver kind of approximate within the right range. We don't have to set an initial guess, but sometimes we get unintended consequences as a result. So even having a guess in the right order of magnitude can do wonders for improving the fit of your model. And so we set those concentrations, and from there, we import these particular solvers from MassPy, and they're specifically the const solver is just this. It's very similar to how the Cobra Pi solver works, except it sets up a problem specifically for solving the concentrations, not the fluxes. And we have an ability to sample those concentrations, and then of course an update model method. And so what we do is we load the model into the solver. We exclude hydrogen and water, and we provide a constraint buffer of 1, which essentially is just essentially the error measurement associated with the constraints. But because we end up solving this problem for the metabolites, we end up formulating it in logarithmic space. We provide 1 here, and that ends up getting converted as well. And so it actually applies in log space. So the 1 works well. Sometimes you need to adjust it. And lastly, for reactions that are at equilibrium and intended to be at equilibrium, we want to let the model know, so it will formulate the constraint appropriately. In the growth data, we had 1 reaction set this way, and that was the LDH reaction, lactate dehydrogenase. And although that isn't typically the case, that the enzyme may be at 0, or it may not always be the case, for this demonstration, we're going to let it stay at 0. And we set this constraint. We make sure that the metabolites that have fixed concentrations remain fixed, and import. And we optimize this problem. And by optimizing it, essentially it's setting up a QP minimization problem in this method, just very similar to the one we saw up here for the fluxes. Just it's developed inside this method. And from there, we can plot these concentrations and look at how they fit. And again, on the 45-degree line means it's perfect fit. And because they're concentrations, we look at this in terms of logarithmic space. As you can see here, some concentrations have a better fit than others, and that will end up being a factor in how some of these models perform later down the road. But for now, we take a look at it. We can compare the initial to the adjusted, and we can also do the same for equilibrium constants, because those are also estimates. And so they get factored into this optimization problem. And we can look at how they are adjusted as well. And when they are adjusted, we can see most of them fit pretty well. We have a little change here for LDH and for FBA, but nothing too major. Lastly, we have to, before we go on any further, we have to fix the metabolite identifiers, as some of them are not SPML compatible. As you can see here, we have some that start with numbers, and that becomes an issue later down the road when we're simulating. So we just fix the metabolite identifiers before we move on. Now, once we've done the concentration adjustments, the next step is to sample around the concentrations. And we do this in this particular workflow, we're doing this to also create an ensemble of models. So the number of models in this particular ensemble is going to be 5, which is rather small, but it will run quickly for this demonstration. And we set up our problem, again, very similar to before, except it's set up sampling problem. And now that it's set up, we adjust the bounds on the concentration variables. And so we get the metabolites from the model, and we adjust the bounds. And again, this problem is set up in logarithmic space, so we have to make sure our bounds are adjusted for logarithmic space. And we're going to allow the metabolites to be sampled within an order of magnitude above and below the initially set value. And then, of course, we solve. Number of samples is equal to the number of models. And we get our samples here. And we can always look at, for a larger sample, we can always look at how the values converge and where they converge to. But for the smaller sample, that's not going to be as helpful. However, the reason we do this sampling step is because this adjustment step right here will find thermodynamically feasible concentrations and adjust the data accordingly, but those may not be the best fit set for simulating them. And so by sampling around those points, we can get a better approximation of which models can perform and find steady states. And that actually leads into this last component, which is another reason we do the sampling, is once we've calculated out metabolites, we need to then, once we've calculated out the metabolite concentrations or sampled them, we then need to calculate the perks for all of these reactions. And we run through this workflow here. And what this is essentially doing is, as it creates the models, and it's going ahead through setting up, grabbing the fluxes, the metabolite samples. It's running S.V to figure out, are there any metabolites that are not balanced in this network? If the metabolite is balanced, not a problem, but if it isn't balanced, it adds a boundary reaction. And the boundary reaction is configured essentially to just allow metabolite to flow into the system freely and flow out, but not to affect the kinetics as best we can. Then we calculate the perks for the reactions. And if there's a negative perk, that indicates that our model was not necessarily thermodynamically feasible in that case, or we maybe had a flipped reaction stoichiometry. But in this case, these models did not produce any negative perks. That gave us an ensemble of 5 models. And just looking at one of them, we can simulate it and see that this one model goes to steady state. And so we'll save this as a reference model to use for analysis later. And that's what this step is doing, is essentially putting this model into a steady state for making sure it simulates to a long period of time. And then if there's a steady state solution, which we know there is to be one, it will update the model and then save the reference model. Now, this is where things get to be a little bit more interesting fun, is we currently have a mass model associated with this, and it's got essentially just the glycolytic reactions and the boundary reactions in it. But what we want to do is we want to add in all of the enzyme modules that we had fitted and created using the MassF package. And so in this case, just to simplify the code and make it a little neater, all of those modules are, or functions to construct, are simplified into this function, and which takes a directory text files created by MassF, and then it imports them and creates the modules. And we also have a couple of isozymes in our system. This is something that we can do to compare the performance of different isozymes. For example, in this case, we have 5 pairs of isozymes for PFK, FBP, FBA, PGM, and pyruvate kinase. However, we have to make an assumption as to how the isozymes handle the flux, because the flux measurements that we have from our data is for net flux again. And so it's not necessarily going to differentiate between the isozymes in this case. So we're going to make the assumption that the major form has about 90% of the flux going through it. And the minor form is carrying about 10% of the overall flux. And that's what this is setting. And then here we calculate out and solve the enzyme modules and put them into our system. Now, something important to note is if the if our reaction stoichiometry is opposite of the enzyme module that we have, we need to flip the reaction flux to be in the opposite direction. And we also need to make sure that isozymes are handled appropriately, since they're not necessarily in our original model. Just depends on the construction of the original model. In this case, IML 1515 does not have them. And so we go ahead and we create the enzyme modules, and this step takes a little bit of time, but luckily, since our ensemble is small, it should go rather quickly. And from here, just to briefly go through what's happening, is it goes ahead, it solves out for all the enzyme it loads out the symbolic solutions that MassF created. From there, it substitutes in our numerical values for our model's initial conditions, which are different for each ensemble and different for each model in the ensemble. It goes ahead, it substitutes in the flux, making sure if it's an isozyme, it multiplies it by the flux split percentage, essentially splitting the flux for different isozymes. If it's not an isozyme, it just puts the flux in directly. We want to make sure that our gene reaction rules get carried through, so our enzymes still have those associations. And then K cluster. Sometimes if you have multiple rate constant sets that can be used for an enzyme, this allows us to vary them. In this case, we just stuck with the first set. And now I've talked through that. We essentially take the reactions that were in our original model. We remove the mass action version, the simplified mass action version of the reaction, and we replace it with the full enzyme module. And that's what happened here. And now it's finished for these 5 enzymes, and it didn't take too long, which is nice. Definitely, again, it depends on the size of the ensemble and the amount of enzymes being computed. And now if we inspect this model, you can see when we simulate it, we end up with all of these our metabolites here, and then all of the enzyme bound forms associated. And something interesting to note is this adjustment that's being made here is actually related to the PGI enzyme, which we had seen at the very top being the one enzyme that did not have a great fit and had some adjustment, had to have its sign switched in the fluxes. So we can save that model as a reference, and we can also save the other models as well. And something to note is our model is no longer a 19 by 12 stoichiometric matrix. It's now 120 by 121 stoichiometric matrix. So adding in enzyme modules definitely increases the size and scope of the model increases the size quite a bit. The scope, the number of initial conditions required, number of parameters. And so what we finally do is we take those models, we just simulate them to make sure that they can reach a steady state. As you can see, all of these reach the steady state, and as a result, we save those models, which we can then use for our analysis in the next notebook. And so I'm going to close my, if there are any questions about that, happy to take them now for this workflow. Otherwise, I'll close this and let Dan flip over to the analysis.
Just curious, have you ever, so you have your integrator tolerances there. Have you noticed if you have to use a different set of parameters for when you start using enzyme modules versus the mass models?
Yeah. So the key parameters that I've had to manipulate tend to be the absolute tolerance, because when you're working with enzyme modules, the concentrations of the bound forms can be much smaller than the concentrations of just the standard metabolites. And so I believe the default tolerance was either 1 to the -9, which may work for some enzymes, not for others. So I just lowered the tolerance a little bit for that particular case. Same with the relative tolerance, although I think in this particular model, the absolute tolerance actually works better than the relative tolerance. And then the other time parameter that sometimes that needs to be set is the initial time step, just because, again, when you add in all of these enzymes, you end up with very stiff systems, especially if you have something like a tetramer like PFK, where you have essentially 4 sites catalyzing the same reaction with the way it's made. And so that creates a very stiff system. And as a result, sometimes you need to add in just an initial time step. And that, let's see. Oh, well, it's not doing my autocomplete in here, but essentially that's the, if you look here, that will be, here we go, initial time step. And usually you just set it to where you want the dynamics to start being captured, the shortest time scale you can. I find that 1e to the -6 has worked reliably well for me in most cases, but that can change.
Got it, thanks.
Mm-hmm. Yeah, I went through this kind of fast, so if there's any other questions about specifics with this, feel free to contact me as well.
Okay, so I can finish this off then. So we just stepped through was the workflow to build an enzyme module model. Very typical with a lot of the debugging steps. So just to reiterate, we were doing a lot of importing real steady state data. So importing in real fluxes and seeing whether with the Qcqa optimization, if we get our model fluxes to match the data. Same with the metabolite levels, bringing in real metabolite levels and seeing if the model can find thermodynamically feasible points that are near the real ones. So these kinds of adjustments are very typical of what we're trying to do when we're starting to deal with real data, and seeing whether the KEQs we import from Equilibrator, which have pretty substantial error, whether any of them have to be adjusted to make the data work nicer together. So this is very typical. And we're always anticipating that there's going to be error in different things, right? There's going to be error in the data. There's going to be error in our kinetic parameters. And so there's always a lot of debugging that goes on as you're trying to find the most consistent model possible. You know that there's going to be error, but you want to minimize that. And so that's a lot of the point of what we're doing. So in this second workflow, I'll step through how do we start to analyze the model properties, and then really do something with it that leads into what you would do with a kinetic model for strain design, meaning namely increasing enzyme concentrations and to mimic overexpression, which is a very typical thing you do when you're trying to increase flux through some target molecule pathway. You're going to be perturbing enzyme concentrations. And you want to find which are the perturbations that are most meaningful. This is an effort that has a really long history. If you're familiar with metabolic control analysis, that was the original goal of that, is understand how flux is controlled. Here we're doing it with simulation because it's just, once you have the model, it's very straightforward to do that. Okay, so we're now just importing in our model. We have a reference model without enzymes and a reference model with enzymes. And we define our isozymes, and we can look at our model properties once again. Stoichiometrics 20 by 28 for the reference model without enzymes, and 120 by 122 for the reference model with enzymes. I don't know if that's the exact same numbers that Zach had, but fairly close. And we can look at these kinds of adjustments. So comparing initial data with computed adjustments. So for fluxes, we're just largely importing this and plotting it just for reference. You can see that my version has a little bit of there's some version issues with matplotlib. Just if you're running this, make sure you use the version that MassPy recommends, because they changed some things afterwards that I think MassPy is in the middle of doing some graphical updates. So everything works, it just throws this warning. And if you want to avoid that, just use the specific version of the libraries, maybe set up a virtual environment for MassPy. But you can see here, we can diagnose whether there are any problematic fluxes, any flux values that the model doesn't like to use. And specifically what pops out is this PGI point here, looks to be set in the wrong direction in the data. So there's probably, so it's going from, it's supposed to be around +5, and it's at -5 instead. Sorry, it tried to be at +5, but it should be at -5. So just probably a mismatch in the flux direction. But otherwise it's the same value. So this kind of procedure can identify things like that, as well as, you know, more fine-grained things like small variation here that can't be perfectly matched by the model. We can do the same thing with metabolite levels. So this is that, again, that result of the concentration optimization. And what do we see here? Well, the majority of the points are along the line, which is good. Let's take a look at some of the off diagonals. So there's one here and one here that, if you look, they're linked, NAD and NADH. So NADH wants to be at, according to the data, it's 10 to the -4. According to the model, so the thermodynamically feasible point is even lower at near 10 to the -5. And it's similar with NAD. The data says it's only slightly under 10 to the -2, but the model says it has to be greater than 10 to the -2. So why is this happening? Is this real, or is this, you know, the model being weird? Well, there are some thoughts that I have on this. One, NAD and NADH are difficult values to measure because there are so many reactions in the cell that are always reacting with them. So in the metabolomics experiment, what you have to do to get a snapshot of the metabolome is to snap freeze your cells. You have to somehow shut down metabolism instantly, because if you just grind up your cells and kill them and like measure metabolism, your enzymes are constantly processing your metabolites in a non-in vivo context and changing those levels away. So you're getting a huge amount of error if you're not able to snap freeze your metabolic state. And so one possible issue is that NAD actually is this high, but after during the extraction procedure, some of it is reacting, and so the measured value ends up being lower, and a lot of it got converted to NADH instead. Whereas what the model says is, in order to run glycolysis effectively, given all the other metabolite concentrations, you really need to have an even higher NAD to NADH ratio, because that's what enables glycolysis to run thermodynamically. So this could be a case where the thermodynamic analysis is actually pointing out a real experimental deficiency. It's possible. It would have to be, you know, tested by varying the extraction procedure and seeing if you exacerbate this issue or something like that. But that's what this would suggest, is that this is actually a meaningful discrepancy identified by the analysis. So that can be nice. KEQs, it's kind of the same story, where you might find some off diagonals and maybe they're more problematic variables. I don't have any particular evidence to suggest that FBA and PYK are particularly problematic KEQs, and they're not even that far off the diagonal. So it's not clear there. But it is known that, I mean, a lot of these KEQs, as I mentioned before, are computational estimated. They can have, you know, order of magnitude error even. So there could be cases where it shouldn't be the metabolites that are adjusting, it should be the KEQs that are adjusting. And we're playing around with optimization procedures that kind of balance the error between these two better to really figure out where the error should be. Like what was the actual problematic data point that gets everything to work out better. So that's very typical of the type of debugging that we'll do. And then we'll adjust the values accordingly to get an internally consistent system and not really worry about, you know, changing the data itself. We just change it within the model. Now, the other key piece that has been extremely useful for us in the debugging process is comparing estimated versus real enzyme concentrations. So when we're defining an enzyme module, we are fully specifying the system almost entirely. We are defining a net flux from flux balance analysis. So all of our V's we have an estimate for. We have concentrations from concentration sampling, thermodynamic concentration sampling. So all our X's are specified. We have all our equilibrium constants from group contribution. And then we have all of our rate constants from mass F. So the only thing that is not specified is enzyme concentration. Now, we could use real enzyme concentrations, but then we'd be specifying everything and things won't be internally consistent within an equation, right? You've over-specified your system. So we actually calculate the enzyme concentration as the last free remaining variable and as a post-processing step compared to the real enzyme concentrations. So this is done for numerical reasons, just so we can fully specify your system at steady state. But it has a debugging application too, because by comparing this, the estimated or the calculated enzyme concentration is how much enzyme is necessary given these concentrations, the metabolite concentration sets, how much enzyme is necessary to catalyze the in vivo flux level, right? So if you need 10 millimole per gram dry weight per hour of flux, how much enzyme do you actually need to catalyze that much based on our kinetic understanding, based on our kinetic model? And then you can compare that to the real enzyme concentration and say, okay, is this realistic or not? If it matches up, then say, okay, great, we have a great understanding of our the kinetics of this enzyme, and it's coming up with a realistic in vivo estimate. If it doesn't match, then you can even use it as a debugging step and say, okay, this is way over or underestimated. What is going on with my model? Why is it not more realistic? And we've used this over and over to debug our models and figure out better workflows. And so I'm going to give you an example of how that works here. So here we have the literature enzyme concentrations, which were from this Heinemann, Matthias Heinemann put out a Nature Biotech E. coli quantitative proteomics study a couple years ago. We just used that. And then here are the enzyme concentrations calculated by subbing in all of these values and the rate constants into the Michaelis-Menten rate laws, or the module, enzyme modules, basically. And this is what it looks like. So we have the literature on the x-axis and the calculated on the y-axis. So you could see that there's some trend here for some of these, but by and large, there's way more estimated than actual for this model. Now, this is not intended to be, a publication, realistic model of glycolysis right now. This is almost debugging for illustrative purposes, but it is very illustrative. So let's take a look at what this actually means. So first thing we notice is that this region is almost entirely depleted, meaning that there are very few cases where the model is much faster or more efficient than the literature. Only this case is very slightly. So by and large, all of the enzyme modules are too slow compared to what they should be in vivo. So they're all up here in this region where much more of the enzyme is needed to satisfy the net in vivo flux than is actually observed. So for example, for PFK, the literature says you only need 10 to the minus 6 PFK in order to meet its net flux. So very efficient enzyme. The model thinks you need over 10 to the minus 2, an extremely unrealistically high enzyme concentration. So something went wrong. Now, what went wrong? Well, that's the model debugging process that we then go through. So we can step through a number of different candidate options. For example, the Qcqa could be too low because in vitro, they didn't identify the best conditions for that enzyme. It could be that the metabolite concentrations are lower than the Km. So the Km could be bad, or the metabolite concentrations could be bad. It could be that an enzyme inhibitor is overactive and is inhibiting this enzyme almost entirely, and therefore is shutting this enzyme down, so you need much more of it. It could be that there's a candidate activator that is combating the inhibitor that we're not aware of. There are all kinds of reasons that this can happen, but our goal as modelers, when we're constructing the model, is to take a look at this plot and then go back to the drawing board and debug these enzymes and try to get them more and more along this line, which would mean that the in vitro model that we have is consistent with the in vivo data. And so that's the process that we've been going through for the tryptophan model. that we did not go through for this initial glycolysis model, which was done really as a demonstration of MassPy's capabilities for analysis. And so that's what I'm going to go through here. Okay, so analyzing metabolites data, let's compare the concentrations when the enzyme modules are present. So we get the metabolite concentrations for the mass model without enzymes. So it's just this pandas data frame, and you just grab the initial conditions for each of the metabolites without it. And then we grab them for the mass model with them, and we can compare them. So we have the metabolite concentrations with the enzyme and without the enzyme. You can see it's really only lactate that changes. The majority of the metabolites are similar between these two models. So maybe only this enzyme was actually affected by the substitution, which seems fine. Then we can take our, I need to actually execute this. I can't remember how far it's been since let me just make sure all this is live. Okay. Okay, got that plot, got this one. Okay, now we can start looking at our ensemble, and now we can do one of our debugging analyses, which is to look at how far are reactions from equilibrium. We talked about this disequilibrium potential. Here, what we're looking at is the mass action ratio, which is derived from, again, the ratio of the product concentrations to the reactant concentrations. So it's, yeah, it's this gamma over KEQ, and then we sub in the KEQ. So so that's just what we're calculating here, is this disequilibrium potential. And we can look at here, and we can plot it. So now what we're looking at is how far is every reaction. We're actually looking directly at the Gibbs energy here, but it shows the distance from it is the disequilibrium. And so now we're looking at the Gibbs free energy of every reaction in glycolysis. And of course, Gibbs energy of 0 means that it's at equilibrium. So the distance here is the disequilibrium. And what do we see? So first off, there are these reactions that are thought to be far from equilibrium, PFK and PYK, that are indeed quite far from equilibrium. So that's consistent with this idea that these are irreversible reaction steps. And that's actually why the model or the cell has evolved alternative pathways around these in fructose bisphosphatase and in PPS, which are like alternative routes around these reactions when you have to run gluconeogenesis instead. And then we see closer to equilibrium are the middle and lower glycolysis enzymes, which is also very canonical understanding. You can see maybe the closest is like PGK here, TPI, thought to be very close to equilibrium. So you have this split in glycolysis between these committed steps, which are highly irreversible. These are also the allosteric sets. And then you have the near equilibrium sets in the middle low glycolysis. Now we can look at, back to the debugging. So what can we do with this disequilibrium potential? Well, one of the thoughts for why enzymes can be inefficient, so in other words, why they would be up here, is if they're very near equilibrium, then that enzyme is thermodynamically very inefficient. It could be catalyzing almost all, almost as much reverse flux as it is forward flux, which could cause the cell to have to produce much more enzyme in order to get net flux. So being near equilibrium is one potential reason why our reactions might be needing more enzyme. So that's what we looked at here, is the concentration of the enzyme versus the disequilibrium. And you really don't see a trend here. So you see this big split where a bunch of reactions are very far from equilibrium and a bunch are close. But it doesn't seem to be very explanatory for why some enzymes are high concentration. You have enzymes that are high concentration and are very far from equilibrium, and you have enzymes that are high concentration and very close to equilibrium, and you know, same on both ends. So this does not seem to be the reason why our enzymes are, some of our enzymes have problematic concentrations. I'll just as I don't go into the exact reason why, but we found that the lack of a KM and slow Kcaps are the main reasons why. And so we've actually started to go back and refit with estimated parameters, and this problem largely goes away. That's something we've applied in the tryptophan model, which is a model that we're trying to use for strain design applications. So So this is kind of the debugging step, but not showing the, what we do after the fact, which is go back to MassF and fit with additional data to make this go away. The other really nice plot we can get is actually seeing what form the enzyme is in. So here we're plotting different enzyme bound states for different isozymes. And so you go to model.enzyme modules, get by ID, and you can get all the different enzyme module forms from that particular enzyme module. And you can sub in the concentrations and figure out what fraction of the total is this enzyme at steady state. So let's look at that for FBA1 and FBA2. So for FBA1, there's 4 different enzyme forms. There's the unbound form, the form binding 2 different substrates, and the form binding both. And same for the different isozymes. And what do we see? We can see that the enzyme is almost none of the enzyme is the unbound form, and almost none is in the double bound form. They're almost all in the single bound form. How is this useful? Well, there's 2 things we can do this. One, we can look across conditions and say, okay, are there changes in these that might suggest what kind of regulation is going on? Like it could be becoming more activated, more inhibited, or just more substrate bound, So you can look differentially across conditions and make calls. Like maybe the absolute value isn't that interesting, but you could look at the differential values across 2 conditions. The other thing we can do is diagnose problematic modules. So sometimes we need an enzyme to be catalyzing flux because we know that it carries net flux, but for some reason its inhibitor concentration is too high or something, and it's like completely inhibiting this enzyme. You'll see like almost all of the enzyme is inhibited. And that can tell you that you have some problematic parameters. Either that metabolite concentration of the inhibitor is wrong, or the Ki is wrong, or there's some unknown activator that combats that, something, right? So it helps you figure out what you should be doing to diagnose your model and improve it later. And you can do the same thing for other inhibitors. This is just showing these different cases. These are all the isozyme pairs. Okay, so just to finish off, let's do a perturbation. So you already saw Zach do a perturbation in his MassPy study and verify that this simulates to steady state. But let's do the type of thing that you would do when using a kinetic model for strain design. So first, we just plot our enzyme at steady state. So we have a nice stable model, no problems there. Now, what happens if we start to perturb our system? So our first perturbation is to add lactate dehydrogenase to the network. And what happens? Well, if you add lactate dehydrogenase, you can watch the lactate concentration. It goes boop, and it increases its steady state concentration. So by adding lactate dehydrogenase, you cause it to produce lactate. It makes sense. It's very intuitive. But you can see most of the network is not changing. You can use MassPy to get more advanced graphics than this, but that's the basics. And here's kind of what you'd like to see is that the LDH concentration initially is going is negative and then comes back to 0, meaning it reaches a steady state again. Okay, now what about overexpression? So this is very typical to what we would want to do when trying to use this for strain design. We want to find that perturbations that affect pathway expression the most, right? We want to increase flux through our pathway. And so you might go through and perturb everything. In this case, we're doing a blanket perturbation. We're doing 2 isozymes by 10-fold and see if anything increases. So we can plot the isozymes themselves, and we can plot FBA, which interestingly ends up decreasing its flux. We can also perturb ATP concentration and see ATP itself go down after initial increase. So it starts high, but then it ends up getting processed and bringing down. And then finally, we can plot all enzymes perturbed at 2 different levels, 2-fold and 10-fold. And just to, again, maybe I'll give a little bit more detail on how to do these perturbations. You just take your initial enzyme. So this is enzyme.initial condition multiplied by 2 in your perturbation parameter of your argument of your sim.simulate function. So the same arguments everywhere, but it's just very easy to implement this type of thing. Perturbation, and now we're perturbing the entire pathway up by 10 then we could see what are the parameters that change the most. So this would be like a blanket overexpression of the pathway trying to increase flux. So does it actually work? Does it actually increase flux? Well, maybe. I mean, it starts out very high, but then it kind of returns down. But it ends up, you can see here, this flux at 10x ends up higher than the flux here. So the y-axes are the same here. You can see it ends up starts much higher, but it also ends higher too. So it does reach a new steady state that's even higher. So this would suggest that the 10x perturbation is bigger than the 2x and has a greater effect on the long-term pathway flux than the 2x. And you can plot even, sorry about the warnings. Here we go. And you can do a deep dive into individual enzyme perturbations and see if there's an enzyme of interest, understand if it's affecting upper or lower glycolysis more, and that type of thing. Okay. All right, so I see that we are out of time, and that's also the last demonstration here. So I can take any questions, but if there's not any questions, then thank all of you for attending, and we will be posting this to YouTube if you missed anything, but feel free to reach out to Zach or myself if you want to get more involved in kinetic or thermodynamic modeling. Okay.
Hey Dan, sorry,
yeah,
I was searching for the button. Just real quick. Yeah, thank you, and also Zach. I think we're actually the only ones here left. Anyways, just one real question. We're talking about the debugging of the model and comparison of measured versus simulated enzyme concentrations. Do you also account for unused enzymes in vivo, like enzyme that is let's say naturally overexpressed and does nothing in the cell?
Yeah, so that's very difficult for the model to know about. You essentially, so that's the same case with these alternative routes through pyruvate kinase. So a flux, for example, in glycolysis, there are these two irreversible reactions in PFK and PYK. And then if you look at enzyme expression, both PFK and its alternative enzyme, like the enzyme that goes in the gluconeogenic direction, FBP, are present in proteomics data. In an FBA model, it will never use FBP on a glycolytic condition because it's just a waste of energy. And so we can't know about that ahead of time because there's no reason for it. So we have to either account for that by approximating how much of it is there or by using proteomics data. But if there's everything that we do in terms of parameterizing the model is meeting the net flux, like finding all the, like what values are necessary to meet the net flux. If there's inactive protein, then we have no idea of how to estimate how much of it is there. However, if you can build an enzyme module, you can still put it in there and set its enzyme as, you could set it at equilibrium, for example, and it won't be doing anything. Or it can be fully inhibited and it won't be doing anything. But that's something that is hard to debug around. You really have to put in all those things hard-coded into the model and kind of hope they're right. We don't have any success cases as far as incorporating inhibited enzymes and being sure that we're modeling them correctly. Right? Really, it's all about the active fluxes and debugging those. Those are the cases where we have like a path forward with debugging them.
Right, because I believe that even for the active fluxes and active enzymes, at least under carbon limited conditions, you sometimes see that you have more enzymes than actually needed. So I was kind of surprised when the enzyme concentrations debugging step that actually your simulations assumes that you need more enzyme than usually say. So I would think that's actually the other way around, but okay.
Right. So inactive enzyme is a thing, but by and large, if we take a look at our enzyme activity plots, the inactive enzyme is always very not present because like in a sense, the model calculates how much of it you need to satisfy the net flux. So if you needed even more, it won't be aware of that. But what I would say is that the amount of inactive enzyme might be, twofold more, right? You might be off by 2, but the degree that we're off in these cases are orders of magnitude. So it's almost like the same. That they are opposite directions of error in that not accounting for inactive enzyme will cause us to be twofold underestimate how much enzyme we have. But this kind of factor of like poor kinetics, poor kinetic understanding or poor inhibitor understanding causes like 4 orders of magnitude off, right? So this just overpowers that other error mode, I think is what we're seeing. But you're absolutely right that should be considered as an error mode, is that these models are kind of maximally efficient, but we know that there's inactive enzyme there. So this won't capture that kind of enzyme just sitting around. This will capture enzyme that's suboptimally bound or enzyme that is inhibited. So there are some methods of inefficiency that are captured by this, but not just like enzyme just sitting around not doing anything, unless it predicts that because like it really comes down to the Km, right? Like if the metabolite concentration is greater than the Km, it's going to be bound, and we'll never get a model that'll say it's unbound. So maybe we just need to understand more about what that unused fraction is actually, what state that enzyme is in. Maybe that should be something that we examine with these models more carefully. That would be good once we solve the bigger issues, or like the more apparent issues, and we get things like lining up closer. Then I think we'll have the resolution to ask that kind of question.
Yeah. Okay. Great. Well, yeah. Thanks a lot for the seminar. Yeah.
Yeah. Thanks for attending.